Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I am your biology educator. So guys, welcome back to Re-NEET Bounce Back series. So today we are going to complete human physiology, which we could not in the previous lecture because of my bad health. So yes, in this part 3 guys, we are going to complete zoology of class 11, that is last 4 chapters of human physiology. Good evening all of you. Give me a thumbs up that audio video is normal and we can start ahead. Yes, please. Quickly let me know. Yes. Perfect. No. Yes. Oh, absolutely. It is normal. Good evening, guys. Good evening. Today, nobody is going to tell me one thing. Ma'am, voice is low. Voice is not low today. Even I am able to shout today. I have bounced back from my sore throat. Yes. So guys, get ready with your NCERTs. Get ready with your NCERTs. We are going to decode every line of NCERT and going to understand human physiology in detail. See, in the previous class, guys, we have discussed 2 chapters. Now, before we proceed, let me remind you once again that you have this great opportunity to watch all the 700 plus lectures, 30 plus tests and 15,000 questions for practice. And you have to take all India mock test series, which will be conducted every Sunday now. So one test is done. I hope some of you have taken that test because I've got that intimation from you. Those of you who've missed it, get ready for the AIMT, that's All India Mock Test Series, absolutely free for next Sunday. Timings would be exactly same, 2 to 5 p.m. Yes, guys. So you can get this Prime membership for just ₹1 on Unacademy Learners app. And also, guys, for those who are preparing for 2027, yes, you have this opportunity to attend our full year lecture under 4,000 rupees only, just by using the code TR Live. And guys, the batches are starting from 20th May. So if you take this batch, you can attend any educator's lecture, mine or any others. I am a part of English batch, and then there are other English batches also. So guys, this is it. And we have already discussed this chapter of human respiration, that is breathing and exchange of gases. Also, we have discussed body fluids and circulation, right? So this is all discussed in the previous class. See, and today guys, we are going to talk about this chapter of excretion. Yes, so we are going to start from here. Let me know that you're all set. So I'll be sharing the notes for previous part, that is complete human physiology part 2 and part 3 together in my Telegram channel. So those of you who don't know what is the Telegram channel, still some of you are asking. So I'll remind you guys, you can log into Tina Maam Biology. This is my Telegram handle and I'll be sharing all the notes here. Yes, guys. So this is my Telegram. And can we start now? Can we start? All right, perfecto. Okay. All right, guys. Moving now to the next chapter. This is excretion. So first of all, just remember that excretory system is that one system which helps us to maintain balance, which helps us in maintaining balance. So we eat carelessly, we take excess of salt and excess of sugar and excess of water, whatever. We just have to like that thing and we go on eating, because of our excretory system, which helps in maintaining the balance in the body. Remember that. So one system that plays major role in maintaining the balance, the homeostasis, the constant internal environment is this system. So see, there are different types of, so this system is going to play 2 important roles. One, removal of excretory waste, and second, maintaining water and salt balance in the body, that is excretion and osmoregulation, right? There are different types of excretory substances produced by an individual, by an organism and by different organisms. But then there are 3 chief nitrogenous waste. One is ammonia, second is urea, third is uric acid. Apart from these, also there are various other nitrogenous wastes. Now see that okay, So we have ammonia, urea and uric acid as the major forms of nitrogenous waste. Now remember, these are all produced as a result of metabolic activities. So all these excretory waste, these are nitrogenous wastes produced as a result of metabolic activities, produced as a result, or we can simply call it metabolic nitrogenous waste. These are metabolic nitrogenous wastes. There are so many, but chief ones are ammonia, urea and uric acid. Let's talk more about them and discuss how they are released from the body. So ammonia is the most toxic. Out of these three, ammonia is the most toxic and it requires maximum amount of water for its removal. So 100%, one thing is for sure, that it can only be produced or released by those organisms who live in water. But I am not saying that all aquatic organisms release ammonia, but all ammonotelics have to be aquatic. I hope you understand this. So all ammonotelics are aquatic but or require water, but all aquatic organisms are not monotelic. Aquatic organisms are not ammonotelic. Please tell me you are clear with this point, right? The second excretory substance that we're going to talk about is uric acid. So ammonia is the most toxic, uric acid is the least toxic. Ammonia requires the maximum amount of water, uric acid requires the least amount of water. Ammonia cannot be stored in the body at all because it is very toxic. Uric acid can be stored in the body for large amount of time. Is that point clear? So least toxic and removed with a minimum loss of water. Please tell me that these points are clear to you all. Is that clear? All right, moving ahead. Next, it says a process of excreting ammonia is ammonotellings. So there are three. Ammonia, so those who release ammonia are ammonotelic and the phenomena is called ammonotelism, right? Urea, ureotelic, ureotelism. Uric acid, uricotelic, CO, right? Urics. So from uric acid we have uricotelic and uricotelism. So these terms should be clear. Yeah, right? So ammonotelism. So what are these examples? Very, very important. So ammonotelism, many bony fishes, aquatic amphibians, aquatic insects. Did I tell you all the ammonotelics require large amount of water? So they are all aquatic. See here, fishes are aquatic. Only aquatic amphibians can do that and aquatic insects can be ammonotelic. Now they require lot amount of water, right? And ammonia is readily soluble in water as well. They're excreted by diffusion across the body and through the gill surface. Cannot be stored in the body at all. So as soon as it is produced, it has to be released from the body surface in the form of ammonium ions. Is this point clear? Right? Now, urea. So after ammonia, it was urea. Urea is in between uric acid and ammonia. It requires water but moderate amount of water. It can be stored but not for a very long time. It is toxic but not as toxic as ammonia, right? So urea is secreted by those organisms who have moderate amount of water for the removal. Now see here, it is a terrestrial adaptation. Very important. It is a terrestrial adaptation. So we needed a lesser toxic form like urea and uric acid, both. So mammals, many terrestrial amphibians and marine fishes, they are ammonotelic. Very important examples. So these are examples of ureotelic. So mammals, we are ureotelic. Then many terrestrial amphibians and marine fishes. So bony fishes, you will remember, bony fishes, A, B, right? Ammonotelic. Ammonotelic are bony fishes. Now here we have marine fishes. Is that clear? And amphibians. So see, aquatic amphibians are again ammonotelic, but the terrestrial amphibians, they are ureotelic. Please tell me that this is clear. Even frog is ureotelic. Yes or no? But frog in the form of tadpole is ammonotelic. Please tell me if you remember these things. Yes, please. Do you remember or you don't? Quick. Extensive question practice, Asha, that is starting from tomorrow onwards. That is starting. By the way, guys, let me tell you, Yavar sir won't be able to take class tomorrow, so we'll have another session tomorrow 7 p.m. and tomorrow we are going to start with plant physiology. So we'll be completing the plant physiology tomorrow at 7 p.m. Okay, so again we'll have a class, so we'll have consecutive classes and very soon we'll be able to complete our class 11. And then after 11, after completing these marathons, right, for 11th and 12th, we'll be practicing lots of questions. I have made my own questions also, which I'll be sharing and practicing with you. And apart from that, PYQs, everything will be happening. We have got this great opportunity to work upon things again. So we are not going to lose this at all. And don't worry about it. Just trust, okay? And have patience. You know, learning demands patience also. And we are, as I've always said, we are not at zero. So keep practicing, and then we'll be practicing here as well and on our special classes too. Okay, perfect. All right, moving on then, moving on. Now see, ammonia, after ammonia we have urea. Ammonia produced by the metabolism is converted into urea. So basically, no matter what you are, whether you're ammonotelic or ureotelic or uricotelic, remember, all different types of organisms, their nitrogenous waste basically is ammonia. How is ammonia produced? By deamination of uric acids, right? By protein, breakdown of proteins. So ultimately, everybody is producing ammonia. But talking about those organisms who are aquatic and they have the leverage to use lot of water, they can release ammonium ions as such through their body surface. But those who can't, their liver converts their ammonia into urea. Now see, this is an important line. Ammonia produced by metabolism is converted into urea. So what does it mean? That ultimately in our body also, the initial step is formation of ammonia. Please tell me these points are clear, right? So liver. So who produces ammonia? It is the liver. Can anyone tell me which blood vessel would contain the maximum urea? Let's see. Can anyone tell me which blood vessel produces the maximum, or contains, not produces, contains the maximum urea? How to join in Phoenix batch without giving Re-NEET? Huh? Nisag. Without giving Re-NEET, you mean you want to prepare for NEET 2027? Renal artery. Asha says hepatic vein. Ureter. I'm talking about the blood vessel. Rajan, I'm talking about the blood vessel. Philip is saying hepatic portal vein. Not sure, right? Okay. Those of you who have said, Jayashri has said this, Asha, and before that, yes, Asha, and then few others who are saying it is renal vein. Absolutely true, because liver has produced it. So the blood vessel leaving the liver will have maximum urea. Yes, beautiful, right? So maximum liver, maximum urea would be found in hepatic vein. Perfect. I will also ask you the other question, but later. What have I written? Hepatic vein, sorry. All right. Is that clear? Now, these animals release into the blood, which is filtered and excreted. Now, some amount of urea is also retained. Now, our body doesn't release all the urea. So, around 17 to 34 grams of urea is normally retained in our blood. Is that clear? That is a normal amount. It has to be retained. Why it has to be retained, you will understand as we proceed in the session. So why is urea retained will be understood, but it has to be retained to maintain a desired osmolarity. Now, where is this desired osmolarity going to play an important role? How is it significant? What does it do? We will understand it all. Now, reptiles, birds, land snails, insects, they are all uricotelic. Now, see, these organisms, they are all dry region. So they have less amount of water. Birds cannot carry lot of water in their body because they have to fly. So again, they have adapted in this mechanism that they release uric acid, because uric acid is least toxic and requires least amount of water. Reptiles, again, snails, land snails, insects, they can't carry lot of water, or they are adapted to dry regions. So they have adapted to uricotelism. Please tell me that these points are clear to all. Now moving on. It says invertebrates. Now there are different types of excretory structures. We are going to talk about excretory structures here. So invertebrates have simple tubular forms, while vertebrates have complex tubular forms, which are known as kidneys, right? We have read about animal kingdom. We have seen protonephridia or flame cells in case of platyhelminthes. This is a unique feature. Then like planaria, etc. Then rotifers, some annelids and cephalochordates like Amphioxus, they all have protonephridia, right? Sorry, they all have this they all have protonephridia. So these are the examples. Then we are talking about nephridia. So nephridia is present in earthworms and other annelids, right? While malpighian tubules are seen in insects, right? Then antennary glands or green glands are found in crustaceans like prawns. Is that clear? While then there are coxal glands also. Coxal glands, we have talked about feather gills, remember, in mollusca. So water vascular system will help in excretion in case of echinoderms. So we have read all about this in our animal kingdom. Now let's come back to, because since we are talking about human physiology, let's come back to human urinary system. So first of all, see, what does human urinary system comprise of? It comprises of a pair of kidneys, 1 and 2. Now do you see that this is the, see, it's like this, right? So this is the right kidney and this is your left kidney. Now our right kidney is slightly lower because on the right side we have liver, right? So this liver on the right side, it pushes the right kidney slightly lower. So right kidney is slightly lower than the left kidney. Is this clear? Then other than kidney, we have these ducts called ureters, which carry urine from kidney to the bladder, which is the third component, and then urethra, which brings the urine outside. Please tell me that these points are clear. Yes, please. Quickly let me know. Yes, guys. So are these all points clear? Please tell me. Perfect. All right. So we have a pair of kidneys. Now see how much these things are present. So this is a pair. This is also in one pair, while this is single and this is also single. Is that clear? Now here, other structures. Can't ignore other structures surrounding the main human excretory system. So inferior vena cava, pelvis, medulla, cortex, we'll discuss it later when we go into the details of kidney structure. So see, above the kidney lies a gland. We are going to discuss about this gland today, adrenal gland. Now renal artery brings blood to the kidney. So this brings the deoxygenated blood, right? And also renal artery contains lot of excretory waste, right? While if we talk about the renal vein will also have, yes, sorry, I've written this wrong. So it contains the oxygenated blood, yeah, and renal vein, it will carry the deoxygenated blood. But what about the excretory waste? So just like, guys, hepatic vein contains the maximum urea, this vein contains the least urea. Yes or no? How many of you understand this point, least urea? How many of you understand this point? Maximum urea was in hepatic vein, least urea will be here because it is bringing that urea here. Urea will be excreted and the blood that goes out of kidneys will contain the least amount of urea. Please tell me these points. Is that clear? Right? So see, this is the inferior vena cava collecting blood from the lower parts of the body, and this of course remains in the lower part, and here is the dorsal aorta and rest of it. Is that clear? The structures mentioned here, other structures we'll be discussing gradually. Now moving on, so this is what they have written. We have already discussed this. So there is this line you must know, kidneys. Kidneys are reddish brown, of course we know. Where are they situated? They are situated between the last thoracic and the third lumbar vertebra. Can I say T12 and L3? This is between, because we know there are 12 thoracic vertebra and 5 lumbar vertebra. So T last thoracic means T12 and L3. Can we remember? And then there is another line. So close to the dorsal inner wall of the abdominal cavity. What are the sizes? 10 to 12 centimeter long, 2 to 3 centimeter thick, and 5 to 7 centimeter wide. Is that clear? This is the weight. Let it go. Now, are these points clear? Let's talk about the structure of the kidney before we see that paragraph. See, we are discussing the structure of kidney. First of all, the protective covering of kidney is known as renal capsule. This is the protective covering. Quickly let me know in the chat, what is the protective covering of lungs and heart known as? Quickly let me know. Let's see how much did you revise. Then guys, overall kidney is divided into two parts. It is divided into two parts. One is cortex and another is medulla. Another would be medulla. Yes. So cortex is going to be the outer part and medulla is going to be the inner part. Please tell me, this is the outer region and this is the inner region. Please tell me. So kidney is divided into two parts, two regions of kidney. Please tell me. So overall, if this is the kidney, we are going to divide this. This is the outer cortex and this is the inner medulla. Are these points clear? Do we see these structures here? Yes. So all this pink structure that you see here, this is all cortex. This is all cortex, right? This is all cortex. Yes or no? And then the structure inner to that. So this whole structure where you see these things, all this is all medulla. This is all inner structure called medulla. Now, do you see that in this medullary region, you see these triangular structures? Yes or no? These triangular structures that you see in the medulla are triangular, right? And that is why we call them medullary pyramids. Pyramids are triangular and they are found in the medulla region. Hence, the name is medullary pyramids. Yes or no? Now, see, inner to this, okay, so here there are funnel shaped structures. The funnel shaped structures present here, these are called renal papillae. These are known as renal papillae. These are the funnel shaped structures below the pyramid, funnel shaped structures. Please tell me these points are also clear. Keep telling me yes, yes, yes. Okay, these are renal papillae. Now, ahead of renal papillae, see, ahead of renal papillae, we have these small ducts. We have these small ducts. So these small ducts, so we have these small ducts. So these small ducts and they gradually open into the large ducts. So these are the large ducts that we are talking about. These are the large ducts, right? Both these small and large ducts are together known as calyces. Calyx means duct. So there are minor, that means small calyces, and then there are major, means large calyces, right? Together these calyces, guys, are going to open in this broad region, and this broad region is known as renal pelvis. Please tell me these points are clear to you all. Please tell me these points are clear to you all, because after this I am going to talk about the sequence. I'm going to talk about the sequence. Yes. So if I talk about the sequence, can I just number it here? It is the renal pyramid opening into the renal papillae, then the calyces, and from calyces it opens into the renal pelvis. And now this broad region, from this broad region arises, see here, from this broad region arises our ureter. From this broad region arises our ureter. Please tell me these points are super clear to you. So this is the fifth part, and ureter will carry it to the urinary bladder. Now listen, do you see that this notch from where the arteries and veins will enter, arteries or veins will enter and your ureter will leave. So this notch-like structure, this notch, guys, is known as hilum. This notch is known as hilum. Please tell me. Do we understand this? Right? So this notch from where the artery, vein are entering and ureter is leaving, that part is known as hilum. Please tell me these points are clear. Yes or no? Yes or no? Now let's go back. Let's go back to this paragraph. What does NCERT say? Towards the center of the inner concave surface. Yes. So this is the center of the inner concave structure surface. We have hilum. Hilum through which ureter and blood vessels and nerves enter or exit. Is that clear? Now, inner to that is the renal pelvis. So we know if we go beyond this, there will be pelvis with projections called calyces. So calyces are nothing but, calyces are the ducts, right? So they have also written the outer layer is a tough capsule, and we understand that capsule is a protective covering. So the kidney is divided into outer cortex and inner medulla, and medulla is divided into medullary pyramids. So these are the medullary pyramids that we have discussed and which project into these calyces. Please tell me that these points are super clear to you all. Yes, guys. Please let me know that these points are clear to you all. Yes or no? Moving ahead. So we have discussed this structure. Now, what are columns of Bertini? Let's go back to this structure. Now, I told you this pink thing, this whole pink thing, this is all cortex. This is all cortex, yes? But do you observe, are you able to see that this cortex is getting in between the medullary pyramids also, right? So this region of cortex going in between the medullary pyramids, this is known as renal column, or we also call it columns of Bertini. In the name of scientist, we can call it columns of Bertini. Please tell me that these points are clear. Yes or no? Please tell me these points are clear. Yes or no? Renal columns. Renal columns are itself known as columns of Bertini. Right? All right, guys. Now see. So we are clear with this? Okay. Now, see, every structure has a unit. For example, lungs have alveoli where the actual gaseous exchange takes place. Now, similarly, kidneys have these structures called nephrons, which are the functional units, and there are around 1 million nephrons in each kidney. It is after each kidney. So we are millionaires, right? So when it comes to the kidney, we are all millionaires. Yes? Quickly let me know that these points are clear. Yes, guys. So is this point clear? Is this point clear? So nephrons, these are the functional units, and each nephron has two parts. Each nephron has two parts. What are these parts? One is the glomerulus, which is a bunch of capillaries entering into the nephron, while rest is the tubular part, and then this tubular part will consist of Bowman's capsule, then PCT, then LOH, then DCT. So these structures are called tubular parts. Please tell me these points. Are these points clear? So what are capillaries? What are glomerulus? These are the tuft of capillaries. Are these points clear? Right? So how are these capillaries formed? Now let's watch that. So let's say this is your Bowman's capsule, which is the first part here. This is the Bowman's capsule, and as we know, it is the renal artery. So this is the renal artery. renal artery is going to break off into so many arterioles, right? And let's say that this is one arteriole. This arteriole is bringing blood towards the glomerulus and we call it afferent arteriole. Are we clear with this? Let's say, so there will be so many afferent arterioles, right? Now this afferent arteriole is going to break off into group of capillaries. So this capillary breakdown, this group of capillaries will be known as glomerulus present in the Bowman's capsule, right? And then it will reform. When it comes out, it is going to reform this blood vessel. This blood vessel will be known as efferent arteriole. So afferent brings blood and efferent takes it away. Efferent takes it away. Now do you see I have drawn this afferent arteriole to be broader as compared to the efferent arteriole? Now this designing, this architecture is going to play a very important role here. Okay, we'll see that this is important for generation of pressure in the glomerulus. Please tell me that these points are clear. Yes or no? So blood from the glomerulus is carried away by the efferent arteriole. It is brought towards the Bowman's capsule by afferent and taken away by efferent. Please tell me these points are clear. Yes, please. Please tell me these points are clear. Yeah? All right. Now let's go ahead. So it says cup-like structure. We have just seen that cup-like structure. And let's discuss more. So this Bowman's capsule, I'm going to draw this Bowman's capsule again. So this Bowman's capsule is a cup-shaped structure, right? Now also it has two walls. Do we see this is the inner wall and this is the outer wall? Remember, the outer wall will be made up of squamous epithelium. Outer wall will be made up of squamous epithelium, flattened cells. This is squamous epithelium. This is your Bowman's capsule outer wall, right? And then this inner wall is going to be made up of special cells. Let's draw them again. So there are special cells here which are like these. So these are special cells present on the inner side, and these cells, guys, are known as podocytes. These are known as podocytes. Okay. Do you see I have left some spaces in between? So these guys, these spaces are known as slit pores. These are known as slit pores, or also known by the name of filtration pores, and through these slit pores or filtration pores are the things going to get filtered. Please tell me that these points are clear. Now, this is Bowman's capsule. This is your afferent arteriole coming. This will be efferent arteriole carrying blood away, right? And in between, let's break it off into a group of capillaries. So this group of capillaries is known as glomerulus. Now, do you see that this glomerulus, guys, and Bowman's capsule is together known as, renal corpuscle or Malpighian body. Please tell me that this point is clear. So what is Malpighian body? Malpighian body is nothing but a collective term used for, glomerulus and Bowman's capsule. Please tell me these points are clear. Right? Do we see this? And then this space in between, by the way, this space in between the outer and the inner wall, this space is also known as capsular space. NCERT is not mentioning here, but the space is empty from where things are going to be filtered. So like this. So that's how things are going to move out, and they will be in the capsular space. Please tell me these points are clear. And after that starts your convoluted tubule, which will be known as PCT. We'll discuss about it in a while. Are these points clear? So this is your NCERT diagram shown here. Do you see afferent arteriole is broad? It is broad, while this is narrow. So what happens? The blood entering is more and the space to leave is less. So this generates, # this generates lot of pressure, generates lot of pressure in the glomerulus. See, whether it's your house or your workplace or even your body, do you see the architecture plays very important role? So we have to read the structure first before we understand the physiology. Generates a lot of pressure in glomerulus, which will be important for ultrafiltration. See, since we are not reading from scratch, so that is why I can use these terms which will be coming in a while. So now you understand why the architecture is like this. Yes or no? Please tell me these points are clear. Okay, right. Now, we understand that after PC, this is your Bowman's capsule. This is your Bowman's capsule. Now, this convoluted structure is known as PCT. Now, this PCT is going to be lined by, this is lined by brush-bordered cuboidal epithelium. Brush-bordered cuboidal epithelium. Why? Because why is brush border present? It is going to increase the surface area for reabsorption. So wherever there is brush border, the surface area will increase. Wherever the surface area is increased, it shows maximum reabsorption. This is a site for maximum reabsorption. We'll see it gradually. And then we have this U-shaped structure called loop of Henle, which will be further followed by the DCT. Okay, now DCT also has cuboidal epithelium, but it is not brush border, because actually in DCT least absorption is going to take place. Is that clear? And then about loop of Henle is the U-shaped structure, LOH. We have two limbs of loop of Henle. Here the filtrate is going to move downward, so we call it descending limb of LOH, while this is called the ascending limb of LOH, because filtrate is going to move upwards. LOH is loop of Henle in the name of scientists. Now, if you talk about this, loop of Henle has two types of cells. It has thin segments lined by squamous epithelium, thin segments, and the thicker segment is again lined by cuboidal epithelium. Please tell me these points are clear. Is that clear? So like this, so many nephrons, they will be together opening in the collecting duct. See, the DCTs of many nephrons open in a straight tube called the collecting duct. And so collecting duct, guys, is not a part of single nephron. Hashtag not a part of nephron. And likewise, this is going to open into the renal pelvis through medullary pyramids and calyces. Are these points clear? So this structure is done. Now, I've already told you that there are two parts of the nephron. One is the outer part is the cortex and another part is the medulla. Now, similarly, see, first of all, let me tell you, where are these structures present? Now, remember, the Bowman's capsule, PCT and DCT, this all lies in the cortex region, while the loop of Henle lies in the medulla. Is that clear? Now, there will be two types of nephrons. There are two types of nephrons. One is the cortical nephrons. So, cortical nephrons means, so I am going to draw this, I'm going to draw this one medullary pyramid. So, this is the cortex region. So, as we see, this is the medulla. Okay. Now, there are two types of nephrons. So, let's say this is the Bowman's capsule, this is the PCT, right? And it has a very short loop of Henle, and then this DCT goes out. Are you getting that? This is one. While there are others in which there is this, again, Bowman's capsule, this is PCT, and then there is a long loop of Henle. There is a long loop of Henle, and again, DCT goes out. Do you see? So guys, the one which a long loop of Henle, right? This nephron will be known as juxtamedullary. These are known as juxta means overlapping, juxtamedullary nephrons. Okay. We just have 15 to 20% of such nephrons, while the majority of the nephrons belong to this category, which is cortical. Do you see it has a very short loop of Henle? Short loop of Henle. Now remember, these cortical nephrons, they form the majority, that is around 75 to 80%. Okay. Okay. Light बंद कर दो. हाँ. All right, are these points clear? Now, what is the difference? Why we needed these two different architectures? See, the long loop of Henle is involved in water conservation. So, it also has a long vasa recta, which is going to play a very important role. We'll be discussing that. While this one will have short loop of Henle and also almost no vasa recta. So, vasa recta is absent here. So it doesn't play any role in water conservation. So no water conservation here. Please tell me these points are clear, right? So we have two types of nephrons here, right? So this is the diagrammatic representation of NCERT as per for the structure of nephron and the blood vessels. The yellow component that you see here, this whole represents the, so this is Bowman's capsule, this is the PCT, and then this is the DCT opening in this collecting tubule, and then in between PCT and DCT we have this loop of Henle. Do you see these structures? Please be very clear with the labelings. Please be very clear with all these labelings. Guys, it is not the time to make friends in the chat, guys. What is happening? Yes, please. Akshay. All right. So see, this is an important line. Vasa recta is absent or highly reduced in the cortical nephrons. Can we move on to the next now? Now see, urine formation is going to require three steps. What are the three steps? One is glomerular filtration, which is also known as ultrafiltration, because it occurs under ultra pressure. Then second is reabsorption, and the third is secretion. Now it occurs in different regions. Let's talk about it one by one. So these three steps, the first one being ultrafiltration, also known as glomerular filtration, because it occurs in the glomerulus and Bowman's capsule. Now see, these data are very important. Now listen, this data 1100 to 1200 ml of blood is filtered by kidneys per minute, and this 11 to 1200 is equal to 1/5 of the total blood pumped by each ventricle, which is per minute. It is saying, it is not the stroke volume. Stroke volume is per beat, and if we multiply it with heart rate, it becomes the blood pumped out by each ventricle per minute. That is the cardiac output, which is equivalent to almost the total amount of blood. So how much blood do you have? Around 5000 ml. Yes, 5000 to 6000 ml. This is the total amount of blood you have. So out of that 5000 to 6000, see, around 1/5, which forms 1100 to 1200 ml of blood, is filtered by kidney every minute. Right? Now for filtration, guys, see here, for filtration there are three layers. Now just in the chapter of breathing and exchange of gases, we have talked about the respiratory membrane. Here we are going to talk about the filtration membrane. Now see, it says filtration membrane. See here, the filtration membrane is formed of three layers, guys. Filtration membrane is formed of three layers. What are these three layers? Let me draw that. So this is the Bowman's capsule. Yes, this is the Bowman's capsule, and I told you this is lined off podocytes. These are podocytes. Yes, these are our podocytes. Remember? And then we have this blood capillary. And the tough group of blood capillaries is known as glomerulus. I'm going to make one of it. So the lining of blood vessels is known as endothelium. Yes or no? We have already discussed it in the previous chapters. This is endothelium. And we also know that the characteristic feature of epithelial tissue is the basement membrane. So do you see that there is basement membrane present in between? Yes or no? So these three layers, endothelium, see here, endothelium of the glomerular blood vessels, yes or no? That is one. Second, epithelium of the Bowman's capsule. See, and the basement membrane, together they make the filtration membrane. So filtration membrane is made up of 3 layers. Do we understand which are the 3 layers? Yes, please. So endothelium, basement membrane, and the epithelium of the Bowman's capsule. And then it is writing here, the epithelium of the Bowman's capsule is made up of podocytes. We have already discussed, and we have read about filtration pores, almost all the constituents of plasma. Now see, now we are coming to filtration. Are the components of filtration membrane clear? Please tell me, are the components of filtration membrane clear? Yes or no? Yes? Now, moving on to the process here. Moving on to the process. See, when I told you, this is a Bowman's capsule. This is the Bowman's capsule. These are the blood vessels, right? This is glomerulus here, and afferent and efferent arteriole. So what happens, since there is lot of pressure, so actually everything is going to come out. Everything present in the blood, plasma comes out. See here, what will not come out? What will come out and form the ultrafiltrate actually, or you call it glomerular filtrate. Glomerular filtrate. Right. What will come out? Everything present in the blood except the blood vessels, the blood cells will not come out, and the proteins will not come out. Rest everything present in the plasma is going to come out. So blood plasma minus proteins. Can I say that? Please tell me. Can I say that ultrafiltrate, this is the composition of ultrafiltrate. So what does it contain? It contains all types of nutrients, glucose, vitamins, minerals, amino acids, then salts, then hormones, complete water. So everything comes out, the nitrogenous waste, of course, everything is going to come out except the proteins. So the day proteins start coming out in urine, now that is an alarming signal. Okay, so this is the component of ultrafiltrate, and this process is known as ultrafiltration. And why do we call it ultrafiltration? Because it occurs under pressure. It occurs under pressure. Why pressure? The first and the foremost reason behind the development of pressure is broad afferent arteriole and narrow efferent arteriole. There are other reasons as well, but we are not going to discuss it as of now. Please tell me these points are clear, right? Okay, moving on now to the next part. So see, the glomerular filtration rate. So the amount of filtrate formed by the kidney per minute. So how much filtrate is produced per minute? So ultrafiltration. So amount produced per minute will be known as GFR, guys. GFR. So amount produced per minute. Please tell me. And how much is the amount produced per minute of ultrafiltrate? Yes. How much is it produced per minute? That is 125 ml per minute. 125 ml per minute, which is equal to ml per minute. And if I multiply it for 1 day, it will become 180 liters per day. Do you understand this? GFR is per minute. Please tell me that these points are clear. The data has to be remembered very clearly. You might get a numerical related to that as well. So these are the data, 125 ml and 180 liters per day. Now this amount will be regulated as per the requirement. So guys, yes, there is a special apparatus called juxtaglomerular apparatus, and remember it is formed by the cellular modifications of DCT and afferent arteriole on the, where they contact. So how it is formed and what are they going to do, I am going to skip it for a while, because in the end I am going to discuss the entire RAS mechanism in detail. Please tell me that these points are clear. Yes, please. So see, the point is the glomerular filtration rate is 125 ml per minute. That means 180 liter of filtrate is produced per day. Now, if 180 liters of filtrate is produced per day, it means that entire day we'll be sitting in the washroom and may not be that as well, because nothing will be left behind in the body. So ultrafiltration is called an unbiased process, because you see it is removing all of your glucose, all of your water. So it is removing both useful as well as non-useful substances. So it's like, you know, cleaning your almirah. When you have made up your mind, I have to clean almirah, you bring out everything, you take out everything except for the hanging rod, right? So you take out everything and then you put back those things which are useful. So now similarly your kidney will be, your nephron will be putting back the useful things back into the blood. Now see, the urine released, the filtrate produced is 180 liter, but urine produced is 1.5 liter only. So that means 99% will be reabsorbed. Now this reabsorption is going to occur in this reabsorption. See, reabsorption is going to occur in PCT, loop of Henle, DCT, as well as collecting duct. Right? And similarly, your selective reabsorption will be throwing out of the non-useful stuff. That will also take place from PCT, LOH, DCT in this. While your ultrafiltration has occurred only in Bowman's capsule and glomerulus. Right? So these are the locations. Now when we are talking about reabsorption, so see, different things will be reabsorbed to different amount and at different regions and by different processes. Please tell me that these points are clear. Please tell me these points are clear. Yes, please. Quick. Let me know that these points are clear. Right? So for example, you see that glucose, amino acid, sodium, they will be absorbed actively. So you need to put energy, ATP will be used, while water and then nitrogenous waste, they will be reabsorbed passively. Is that clear? Is this point clear? And then tubular secretion, where still there are certain things left in your blood which needs to be pulled out. So it has to take place actively. So remember, tubular secretion is also an active process. It is an active process. In fact, those organisms which do not have Bowman's capsule and glomerulus, in that case, ultrafiltration doesn't occur. So in those organisms, what will happen? Tubular secretion is the only method of removal of nitrogenous waste. Only method of removal of nitrogenous waste. Please tell me these points are clear. Right? And then talking about the different regions, I have already told you PCT has brush bordered epithelium, increases the surface area and nearly all the nutrient. Why? Because maximum reabsorption has to take place, and it is writing 70 to 80% of electrolytes and water are reabsorbed, and it is a place, I've just told you, where tubular reabsorption as well as selective secretion will take place. Then talking about loop of Henle, this is the place with minimum reabsorption, especially in the ascending limb. Okay, and it is going to play important role in maintenance of high osmolarity. How, when, why, we will discuss it. Descending limb, okay. Now see, if we talk about the loop of Henle, I'll tell you right here about the nature of descending limb. So this is the descending limb of loop of Henle, while this is the ascending limb of loop of Henle. Right. So basically, what's going to happen? For descending, so from descending, water can come out, right? And from ascending, sodium can come out at the expense of potassium. So here, sodium-potassium pump takes place. Sodium-potassium pump is active. So basically, ascending limb is permeable only to sodium, while descending limb is permeable to water. Can we say that? The descending limb is permeable to water, do we see, and almost impermeable to electrolytes, while the ascending limb is impermeable to water and allows the transport of electrolytes actively. We must know that sodium-potassium pump is an active pump. Remember the nerve conduction? Yes or no? Please tell me. Yes or no? Do we remember things? Yes? All right. Moving on now, moving on to the DCT. So we know that DCT, see, maximum absorption, PCT, minimum in loop of Henle. If we talk about DCT, it is conditional only if required. So we also call it facultative. And this facultative, that means conditional only, it will occur under the influence of some hormones. It will occur under the effect of certain hormones like ADH, right? So we'll see that getting applied here. So both absorption and secretion will take place. So this diagram summarizes everything basically. So see here, this is your blood vessel. So from your blood vessel, things are moving into the nephron. So this represents, this represents your tubular this is tubular secretion. This is tubular secretion. And again, this is your blood vessel. So things moving out of the nephron, this represents your tubular, reabsorption. Yeah, this represents your reabsorption. So reabsorption is nephron into the blood, right? Now can you see that this part is PCT? So things written here are being absorbed and secreted through PCT, and this is your DCT. And then water moving out of the descending limb we have just seen, and sodium chloride moving out through the ascending limb. Can we note it down? Also, do you see that urea, we have been mentioning again and again that urea is not completely removed from the body, it is retained in the body. So do you see that collecting duct, this is the collecting duct will release some urea into this space. So the space around the nephrons in the medullary region, this hole, so see, this is all medulla, right? This is known as medullary interstitium or interstitial fluid, right? So there is lot of urea present here. So there are two compounds which are going to play very important role in maintaining the osmolarity of medulla. So one is sodium and second is urea. They maintain the osmolarity here in medulla. How that is going to play important role, we will see it. How that is going to play important role, we are going to see it. Is this point clear? Yes. Now, collecting duct, I've told you it is not a part of one nephron, right? It extends, and we've also seen that PCT, Bowman's capsule and DCT, they lie in the cortex, while a long loop of Henle lies in the medulla. But if we talk about the collecting tubule, it extends from cortex to the inner parts of medulla, right? And large amount of water could be absorbed from this region. Again, this absorption has to be facultative or conditional. It is your ADH that is going to absorb this water. Is that clear? And then selective absorption of H positive and K positive ions. Now remember guys, it is only mammals who have this unique feature of concentrating their urine, and in order to concentrate the urine, they have a very special architecture again. And what is that architecture? They have this U-shaped loop of Henle, and along the loop of Henle we have some blood vessels. So the tuft of blood vessels in Bowman's capsule was glomerulus. Now similarly, we have a specific tuft of capillaries around the loop of Henle, which will be known as vasa recta, and together vasa recta and loop of Henle are going to play this important role in concentrating the filtrate or concentration of your urine. And now only mammals have this ability. Now first, and this mechanism guys is known as counter current mechanism. This is known as counter current mechanism. Now let me tell you the word meaning of this first. Counter means opposite, current means flow. Now we will see that there are things flowing in the opposite direction. Like how? See here, I'll draw the loop of Henle first. See, this is your loop of Henle. This is the descending limb, right? This is the ascending limb. This is loop of Henle. Now, surrounding the loop of Henle, I told you there are blood vessels. There are blood vessels, and these blood vessels are known as vasa recta, right? Now, what is counter current here? Now, what is counter current here? So, this is descending loop of Henle. We know the filtrate is flowing in the downward direction, and in this loop the filtrate is flowing in the upward direction. Do you see it is counter current? So between the two limbs of loop of Henle, this is one. So counter current is operating at 3 levels. This is operating at 3 levels. One between ascending and descending loop of Henle. Yes. Similarly, you will see this vasa recta is bringing the blood downwards, and this vasa recta will carry the blood upwards. So I will call this limb as descending vasa recta, and I'll call this as ascending vasa recta. So the second level at which counter current is being maintained is ascending and descending vasa recta. Do you see the two levels at which this is being operated, right? And now, because the two loop of Henle and vasa recta, they are in very close association, they are in vicinity to each other. So see, the filtrate here in descending limb is going downwards and the blood is going upwards. So do you see a counter current operating here? Filtrate down, blood up. And similarly, here filtrate up and blood down. So between the adjacent limbs of loop of Henle and vasa recta, do you see that counter current is getting operated at 3 different levels? Please tell me yes or no. Maybe to maintain osmotic, not maybe, Nandini, 100% it is. Is the voice lower? 100% it is. No. I've told you. Don't tell me that. Is it only Hanuman who cannot hear me properly, or is there a voice problem? Please tell me. Please tell me, guys. Is there a voice problem today? I don't think so. Audible and it is crystal clear. Increase. Who is saying increase? Yes? Perfect. Thank you. Alright guys, now see, now how is this counter current going to help? So these are certain lines written here, but we'll be able to understand it only in a while. Now see, this is your NCERT trying to show the counter current mechanism. Do you see? They have shown this as a loop of Henle, descending limb and ascending limb, and this is your vasa recta. Right, but I always make my own diagram to explain this, so let's get to that. So see, this is your loop of Henle. This is the descending limb of loop of Henle, because filtrate is going to move downwards, and this is your ascending loop of Henle because the filtrate is going to move upwards. So you see the counter current operating here. Similarly, very adjacent to that and near to that is your loop of Henle here. Sorry, your vasa recta. Right. So here the blood is going to move downwards, so we call it descending vasa recta, and the blood is going to move upwards, so I'll call it ascending vasa recta. Yes or no? Right. Now remember, when the filtrate enters the loop of Henle, now please pay attention, when the filtrate enters the loop of Henle, the osmolarity is around 300 milliosmoliter. Is that clear? What is the osmolarity like? 300 milliosmoliter. Also, let's not ignore the surrounding interstitial fluid. This is the medullary interstitium. Medullary interstitial fluid or interstitium. Is that clear? Also see that in this interstitium, as we go from outside to inside, deep into the interstitium, the osmolarity will also go on increasing, like 600, like 900, and maximum it goes up to 1200 milliosmoliter, osmo per liter. Is that clear? Now, this is what we have read. What is majorly responsible for maintaining this osmolarity? Who is responsible? It is sodium chloride and urea. So how has the sodium chloride and urea entered into the interstitium? We'll understand it here. Now see, I'm coming back to the descending loop of Henle. So when descending loop of Henle enters, the osmolarity of the filtrate, guys, is 300. How much is it? 300. Now see, what will happen? We have read that descending limb is permeable to water and almost impermeable to electrolytes. So water is going to come out. Water is going to come out. Where? This is all interstitium. This is all interstitium, right? This is all interstitium. So what is going to come out? Water is going to come out. Yes. So when water will come out, what will happen? The filtrate in the descending loop will become concentrated. You are removing water, the osmolarity will go on increasing, and it increases up to 1200 milliosmo per liter. Yes or no? Now, as soon as the filtrate enters the ascending region, the osmolarity changes. Now, instead of losing water, it starts losing sodium. It starts losing sodium. So, see, this is how sodium came out, right? So, sodium is getting lost from here by using ATP, that is active transport. Sodium will be lost, potassium will move in. Please tell me this point is clear as well. So, when the osmolarity was 1200, the loop of Henle started removing sodium. So, if you are removing sodium, what will happen? It will again become dilute. So, the osmolarity starts decreasing again. Yes or no? Please tell me, till it becomes 300. That is the normal osmolarity of our body. Please tell me that these points are clear, everyone. Is that clear? Okay. Now, what will happen? Now, see. The water that has come out, it is all in the interstitium. The sodium that has come out is all in the interstitium. Now, there is a vasa recta in close association with the loop of Henle. So, the sodium which has come out starts moving into the blood. This sodium starts moving into the blood. Now, see, the blood which was coming here also had osmolarity of 300. Now, when you will start adding sodium to this, what will happen? Osmolarity will go on increasing from 400, from 600, from 900, and it will go up to 1200. Why? Because you are adding this sodium coming out from loop of Henle is getting added into the blood. Yes or no? Actively. Is that clear? So, now the sodium is getting added into the blood. Yes. Now what will happen? The blood which is coming from here, which is near to ascending loop of Henle, is absorbing lot of sodium. Now as soon as this blood reaches here, it will try to pull out water because it is releasing water. It is, see, this blood is thirsty now because it has lot of salt. Now this blood wants water. Now this blood wants water. So the water that is coming out of this is going to move into the blood. This water is now going to move through interstitium, of course. Through interstitium, it is going to move into the blood. Yes or no? Please tell me. So the osmolarity of the blood was 1200 here, and after absorbing water, it will start decreasing to 600 and 400 until it becomes 300. Please tell me these points are clear. So, descending limb is losing water. Therefore, guys, descending limb is known as, it is diluting water, it is concentrating limb. It makes the filtrate concentrated or hypertonic. While this is losing sodium, it is making the it is making the filtrate dilute. So, this is the diluting limb. It makes the filtrate hypotonic. Please tell me these points are clear. Yes, please tell me. So guys, this interchange of things between the loop of Henle and vasa recta is going to concentrate your urine. Yes or no? So see, this is going to form urine. So the urine has lost water. It has gotten concentrated. It has lost sodium also. So sodium has been brought back to the body, which was actually just filtrated by a glomerular filtrate. Do you see that? No doubt PCT absorbs the maximum water and maximum sodium, but it is Mammals have additional counter current mechanism to concentrate it even more. Please tell me these points are clear. Please tell me these points are clear. Yes or no? Can we proceed? So guys, that was the major thing. So humans can produce urine nearly 4 times. Why is it saying nearly 4 times? Because see, when glomerular filtrate is produced, the osmolarity is around 1200. Okay, now PCT is going to make it around 300. So when it enters, it is 1200. So when filtrate reaches the loop of Henle, it becomes 300. So basically, PCT makes it isotonic to the blood, because blood has also 300 osmolarity. So it makes it isotonic. So it loses water, makes it hypotonic. It loses sodium, makes it hypotonic. Is that clear? Please tell me these points are clear. So finally, when the urine will be produced, let's say if conditional absorption also doesn't take place, so urine will have roughly 300 osmolarity, and the initial filtrate formed had osmolarity of 1200 milliosmo. So how many times has it been concentrated? It has been concentrated up to 4 times. Please tell me if these points are clear. Yes or no? So human kidneys can produce urine 4 times concentrated by the initial filtrate form. Please tell me these points are clear, right? And finally guys, the regulation. Now this is very important. So regulation is there are 2 types of regulatory mechanisms that we are going to see. One is the RAS mechanism and second is the ANF. So RAS mechanism operates when your blood pressure or GFR falls, and this operates when your BP becomes very high. So this is going to increase the blood pressure gradually, and so it is going to increase the blood pressure. So this operates when BP is high, so this is going to lower the blood pressure. So let's see how these things happen. So remember, first thing, osmoreceptors are present in the body, and these osmoreceptors can they can get activated when the changes in the body fluid volume or ionic concentration takes place. Excessive loss of fluid will activate the receptors, which will stimulate this. I have underlined the important lines. I'll be sharing these notes on the Telegram channel. Those of you who have missed the Telegram channel, guys, my Telegram channel is Tina Maam Biology, and I'll be posting all the notes here. And also now listen, what has NCERT said? Let's see. So guys, when the blood pressure or the glomerular filtration rate falls, this is detected by the osmoreceptors. Right? Now these osmoreceptors are going to send signal to your kidney. So there are some cells known as, there is a special apparatus known as juxtaglomerular apparatus here. Is that clear? It stimulates your juxtaglomerular apparatus. Now this juxtaglomerular apparatus is going to secrete renin. So who is going to secrete renin? It is your kidney. Renin is a hormone. So this hormone is being secreted by your kidney, and it will be put into your blood. Now see what will happen in your blood. So this is your blood. So your blood already has angiotensinogen. Now this angiotensinogen is in the inactive form. When renin enters, this renin is going to activate this angiotensin into angiotensin 1. So this will be converted into angiotensin 1 on stimulation of renin, which comes from your kidneys. Now, by the way, how did angiotensin got produced? So it is your liver. Which had produced it and put it in your blood, but in the inactive form. So as soon as the renin enters, it converts it into angiotensin 1, but angiotensin 1 is further converted into angiotensin 2 by the presence of another enzyme, which is contributed by your lung capillaries, and we call that ACE. ACE is angiotensin converting enzyme, comes from your lungs. Is that clear? Now, once angiotensin 1 is formed, now see, it is a game changer. This angiotensin 1 is going to have three-dimensional effects. Number one, it is going to stimulate your hypothalamus. So, hypothalamus will further stimulate, it is going to stimulate your hypothalamus, and hypothalamus will release or produce through posterior pituitary, it is going to release ADH. It is going to release ADH, antidiuretic hormone. Right? Now, what does this antidiuretic hormone do? Antidiuretic hormone is going to reabsorb water. It will reabsorb water from DCT and collecting tubules. Please tell me these points are clear. If water will be reabsorbed, it will be put back in blood. What will happen? The blood volume will increase. If blood volume will increase, the blood pressure will increase. And this is what we want because the blood pressure has fallen. Right? Now, what is the second effect? The second effect would be angiotensin 2 is a potent vasoconstrictor. Look at the name, angiotensin. Angion means blood vessel, tension means pressure. So it is a potent vasoconstrictor. So when your blood vessels are constricted, area decreases, pressure increases, it itself increases the blood pressure, yes or no? Ultimately, this is also going to increase the blood pressure. And then the third effect that it will have is on adrenal cortex. Now, adrenal cortex gland, as you know, secretes mineralocorticoids like aldosterone. And this aldosterone, guys, is going to reabsorb sodium, conditional reabsorption of sodium from DCT and CT. Right? This is also from DCT and CT. It will increase the osmolar permeability of water in DCT, ADH. Now, do you see? Aldosterone is going to absorb a lot of sodium, and if sodium is reabsorbed, ultimately water will also be reabsorbed. More water reabsorption will take place. More water reabsorption means what? More blood volume. Ultimately, this is also going to increase the blood pressure. So, do you see that there are three main heroes here? There are three main heroes here, which are renin. This is renin. Second is your angiotensin 2. Third is your aldosterone. Right? So, these three, we call it RAS mechanism. S for system. RAS mechanism. So, this RAS mechanism is stimulated when your blood pressure or GFR decreases. Please tell me that these points are clear. Yes, guys. Please tell me these points are clear. And now, opposite to this, guys, we have ANF. So, when your blood pressure increases or GFR increases, again, it is detected by the osmoreceptors, right? And then they send signal to atrial walls of the atria of your heart, atrial walls, right? Now, atrial walls are going to produce, these atrial walls are going to secrete ANF, natriuretic factor, AANF, and this natrial factor, ANF, going to counter the RAS mechanism. It stops the RAS mechanism, and therefore your blood pressure is going to decrease. This is what we want for now. Also, this ANF is going to dilate your vessels, vasodilation. Again, vasodilation is going to decrease the blood pressure. Please tell me that these points are clear. Please tell me these points are clear. Yes? So guys, with this, we complete the regulation. So this is what has been written, and this is what I have already explained. You should know the whole process in the correct order, right? Fall in this. Activation of JG cells converts angiotensin into angiotensin 1, then converted to angiotensin 2. Step 4, angiotensin 2 is a vasoconstrictor, increases this, activates cortex through aldosterone, reabsorption of sodium, and water from DCT, and this is angiotensin, and then this is ANF. It causes causes vasodilation, and it is a check on RAS mechanism, and thereby decreases the blood pressure. Please tell me these points are clear. Quick. And then we have micturition, guys. What is micturition? Micturition is release of the urine from bladder. So how does this whole mechanism start? So see, urinary bladder stored till a voluntary signal is given by the central nervous system. Signal is initiated when the stretching of the urinary bladder takes place, because they have stretch receptors. So from the stretch receptors, message goes to the central nervous system, and central nervous system in the walls of bladder. So from the stretch receptor, signal goes to central nervous system, and central nervous system is going to send signal through its motor nerves to two places. Where are the two signals going to be sent? One, the smooth muscles of bladder, and second, for the relaxation. So there will be two effects. Your contraction of bladder will take place, and in order to evacuate the bladder, relaxation of sphincters have to take place. Relaxation of sphincters have to take place. Please tell me these points are clear. Right? And this whole neural signal coming from CNS to the bladder for the release of urine, this is called micturition reflex. And how much urine do we produce per day? 1 to 1.5. And then yellow colored watery fluid, slightly acidic, pH is 6, around 6, characteristic odor. How much urea do you release per day? That is 25 to 30. Analysis of urine can actually give you an insight into the health of the kidneys. So if you see the presence of glucose or presence of ketones, that is a signal of diabetes mellitus. So glucosuria, you can also write it glyco or glucosuria or ketonuria, is an indication of diabetes mellitus. Now there are other organs also involved in excretion. One is your lungs. Second would be your second would be your liver. Third would be your sweat glands. Fourth will be your sebaceous glands, right? And also your saliva. So these are the other structures. So talking about lungs, they release around 200 milliliter of carbon dioxide per minute, and also, you know, when you exhale, water vapors are also lost. Talking about liver releases bilirubin, then some steroid hormones, cholesterol, biliverdin, vitamins and drugs. Talking about sweat, along with water, it is sodium chloride and some amounts of urea also that comes out through your sweat. Then lactic acid as well. Sebaceous glands are your oil glands, and what do they release? They release sterols and hydrocarbons and waxes along with the oil, and otherwise this oil forms a protective covering for your skin. And that it says, do you know there are small amounts of nitrogenous waste even in your saliva? So yes, we do release some urea in our saliva as well. Please tell me that these points are clear. Right? Now last not the least, we have some disorders. So as I have already told you, some amount of urea is retained. So around 17 to 34 gram, that is considered normal. But if more than that urea is retained in blood or is not being removed by the kidneys, that condition is known as uremia. Accumulation of urea is called uremia, and it is an indication of kidney failure. So when the kidney is failing, either you get a transplant or you wait for the right donor. Till we are waiting for the right donor, we have artificial kidney or the hemodialysis, right? So what do? We take out blood from a convenient artery, put it in the dialyzing unit, right? So we put, before putting it in the dialyzing unit, we put anticoagulant, and then we put it in the dialyzing unit, which has lot of cellophane tubes, right? So now this is very important. So dialyzing fluid, the composition of dialyzing fluid is same as plasma, except in plasma we have some urea, but in dialyzing fluid we don't have urea. So what will happen? So because it is a blood of a person whose kidneys are not functioning properly. Now see here. So this is the dialyzing fluid, and there is lot of dialyzing, this is the dialyzing chamber, sorry, and this is the dialyzing fluid all around. Is that clear? So we take out blood from an artery, we add heparin to it so that the blood doesn't clot when it comes out, and from here it goes into these special tubes present within this chamber. Right? So these tubes are made up of special membrane known as cellophane membrane. So what happens? These are cellophane tubes. Now this is the dialyzing fluid. Okay, dialyzing fluid. Now dialyzing fluid is very similar to our plasma, just that it does not contain urea. No urea in this. Now see, when we run blood through this, then there is blood running through the cellophane tubes, blood of that person whose kidneys are not functioning properly. So it means this blood is going to contain lot of what? It contains a lot of urea, high levels. And there is no urea in the dialyzing fluid. What will happen? Urea will come out. So urea will come out because of from high concentration to low concentration. Simple as that. Dialysis basically means diffusion. Okay, so urea is going to come out and the blood is getting filtered. So here there will be low urea, and now this blood can be put back by adding anti-heparin. This blood is pumped back into the body through a vein. Is that clear? Now what happens? This is how the blood is cleared out of cleared for urea. Now what happens here is, now while this urea is coming out, the urea is getting collected in the dialyzing fluid. So you have to keep on changing this dialyzing fluid. So new fluid is entered and the previous one or the used one is removed. Because if the level of urea is increasing in the dialyzing fluid, it can also go in the reverse direction back into the blood. So you have to take care of this. Please tell me these points are clear. Please tell me these points are clear. And then once you get the suitable donor, which can be your donor can be a close relative, then you go for the kidney transplant. Is that clear? Why a close relative? Because then your immune system is also doing its part, right? So to decrease the chances of rejection, the graft rejection. Now, renal calculi are the stones in, and majorly they are the crystallized salts of oxalates, right? And then glomerular filtrate, it is the inflammation of glomerulus. Please tell me these points are clear. Let's have a thumbs up break. Quickly let me know, guys, that we are thorough with this chapter. We have read it from zero to hero level. And once again, I'm going to repeat that tomorrow also, guys, we are going to conduct a class, this again marathon of 3 to 4 hours, and we are going to complete the plant physiology tomorrow 7 p.m. because Yavar sir is not available and I will be taking your class. So botany part 1 of class 11. So we'll be starting from plant physiology and then I'll be going on to, in the next part, we'll be discussing plant kingdom and biological kingdom and cell unit. Please tell me these parts are clear. Quick. Yes? Perfect. Have you all liked the session? I can't see so many likes. There are more students watching and less likes in the class. Not fair, guys. Less likes. Uh-oh. When will you complete the syllabus, ma'am? We are discussing all the things in detail. See, it's not a long, I think I've hardly joined 15 days back before your 3rd May exam. So I had just few days to complete the syllabus, right? So I did it in hurry also, but now I have time, we have 1 month and we are going to take just how many sessions? We are going to complete our syllabus in 8 to 9 sessions. We'll be taking these marathons and completing the syllabus in detail, right? That is very important. When God has given us this opportunity, let's make good use of it, okay? Let's not keep on crying now, what about whatever happened to the system. So now we are going to make the best use of it. So yeah, it will take around 8 to 9 sessions. And from tomorrow, I am going to discuss your plant physiology. And yes, 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 yes. So yeah. And thereafter, once we complete the syllabus, we'll be discussing only the key points. I'll be giving you lots of cheat sheets. I'm going to discuss lots of questions, my own made questions also. We'll be discussing it all. Right? I don't speak Telugu. Dhanushri Shreddy. No, I don't speak. I speak Punjabi, I speak Hindi, and I speak English. Okay. Chalo. Let's start with the next chapter. Break over, guys. Break over. And let's move on to the next chapter, movement and locomotion. See, starting with the important words. So, streaming of protoplasm. This is a basic type of movement seen in organisms like amoeba with simple type of movement. Then there are other movements like cilia, flagella, and tentacles used by many organisms. Chance of, so basically, why does locomotion occur? See, I hope you know the difference between the two terms. Locomotion is when there is a change of place, right? And movement can be while you are standing at one place also. Like while I'm speaking, my tongue is moving, eyelids are moving, right? So all movements, see, this is a line, all locomotions are movements, but all movements are not locomotions. So the point here is, why do we move? Why do we change our place from one place to another? So the change of place or location is, guys, in why do we do that? This is always true. It is written here. Locomotion is generally for the search of food, shelter, mate, and suitable breeding grounds or favorable conditions, or to escape from the enemies. So this is the motive behind the locomotion, and this locomotion is always voluntary. Talking about paramecium, it uses cilia, while if we talk about hydra, it has tentacles for its movement. Is that clear? So overall, there are three major types of movements. So although NCERT writes three here, but in the next paragraph, it is going to mention about the fourth movement also, which is going to be flagella, right? So overall, guys, we have four different movements basically. So amoeboid, ciliary, muscular, and flagellar. Let's discuss it one by one. So where are the amoeboid movements seen in our body? So amoeboid movement means amoeba-like, where pseudopodia is put to use, right? So amoeboid movements are seen in case of macrophages and leukocytes, that is your WBCs, where they show amoeboid movement and perform diapedesis. So they can change they can squeeze themselves through the blood vessels. Amoeba doesn't have a fixed shape. It can squeeze through anything. Now, similarly, your WBCs can squeeze through anything and show diapedesis. Are these points clear? Please tell me. Are these points clear? Yes or no? All right. Now, it says, so how does amoeba move? Using the pseudopodia, which helps in the streaming of the protoplasm, and now the cytoskeleton elements and microfilaments are also involved in the amoeboid movement. Then talking about the ciliary movement. So paramecium-like organisms, they use cilia, but what about the ciliary movement in our body? So ciliary movement is seen wherever ciliated epithelium is present. Where is ciliated epithelium present? We have discussed in trachea, bronchi, bronchioles. So major part, most of the part of your respiratory tract has ciliated epithelium. Why? Because it has to push the mucus upwards, right? So see here, and also into the internal tubular organs. For example, trachea for removing the dust particles and foreign particles, and then passage of ova through the FRT is also facilitated. So basically, your fallopian tubes are lined by ciliated columnar epithelium, right? And your trachea is lined by ciliated pseudo stratified epithelium. Please tell me that this point is clear. Please tell me these points are clear. Yes or no? Please tell me these points are clear to everyone. NCERT says it is cuboidal, right? Sorry, not cuboidal, columnar only. So it is ciliated epithelium. right? Then talking about the third type of movement, which is muscular movement, which is seen in limbs, jaws, tongue. And now the muscular movement has a unique property which is not seen in any other movement, and that is the contractile property of muscles. It is this contractibility that allows movement. Now this is an important line. It is already a PYQ. Please focus here, that locomotion requires a perfect coordination of three different systems. One is your muscular system, second is your skeletal system, which comprises of bone and cartilage, and third is your neural system. So impulse, your message has to come through neural system, and then the muscles attached to your skeletal region or skeletal system are going to help in locomotion. So three systems which are involved are these, right? And also guys, we have to talk about three different types of muscles. What are the three different types of muscles? What are the three different types of muscles? Yes, so we have striated muscles, also known as voluntary, also known as skeletal because they are attached to the bones, right? Then there are smooth muscles. Smooth muscles are known as visceral muscles because they form the internal structures. They are not related, no, sorry, they are not striated, so we call them non-striated. And since they are non-striated, they appear smooth under microscope, so we call it smooth muscles. And then we have the third type, which is the cardiac muscles. Yes or no? Right? So I'll draw the structure here. How does striated muscles look like? So striated muscles look like this. So they are cylindrical, they are multinucleated, and they occur in bundles. So this is how your skeletal muscles look like, right? These are your skeletal muscles, cylindrical. So cylindrical, they are not branched, they are unbranched. They are multinucleated, we call it syncytium. And syncytium, and we'll see the detailed structure of this muscle. It will show some striations here. It will show some striations, hence the name striated. And how is the shape? It is cylindrical, and these cylinders are going to form bundles, so they occur in bundles. So cylinders form bundles here. So these are the properties of your cylindrical muscle. Now talking about your non-striated muscle. Non-striated muscles look like these. So they are spindle shaped, they have a central nucleus. Also, by the way, here syncytium, and they have a peripheral nucleus. That is, nucleus is towards the periphery. Peripheral nucleus. While if we discuss the smooth or the visceral muscles, how is the shape? The shape is spindle shape. Spindle shape means they are pointed at the ends and bulging from the center, tapering at the ends, right? Tapered ends means pointed ends. Then they are also unbranched. Spindle shape, unbranched again. Now they were multinucleated, so we call them syncytium, while they are uninucleated, and the nucleus is right in the center. Uninucleated with central nucleus. Do we see that? Now they don't have any striations, so no striations, hence they appear smooth. No striations. They are not cylindrical, they are, okay, so I've already written this, sorry. So they are not cylindrical, they are spindle shape, and they don't form bundles, they rather form sheets. They form sheets. Clear to all? And then guys, we have cardiac muscles. Now remember, cardiac muscles resemble to smooth muscles when it comes to the activity. Heart muscles are not under your control, they are involuntary. So function wise they resemble the smooth muscles, while structure wise they are more or less like striated. Why? Let's see it here. See, so cardiac muscle fibers, they are cylindrical like striated muscles. They have faint striations, they have faint striations like this. Not as many as these, but they still have faint striations. But unlike striated, they are branched. So they are branched like this. Is that clear? So they are branched like this. Okay, but This is multinucleated syncytium. This is uninucleated. There is single nucleus here. Okay. Now, unique feature of these muscles, very unique feature of these muscles are, wait. So, these muscles are, they have intercalated discs which are not found anywhere else. They have intercalated discs. Intercalated discs. Please tell me that these points are clear to you all. Yes, guys, please tell me these points are clear. Hanji, all clear? So now see the structure. How is this? This is branched. This one is branched, right? This one is uninucleated. This one is cylindrical. This one has, this has one nucleus, right? And they also form bundles. They also have faint striations. Please tell me all these points are clear to you all. And they have special intercalated discs. Now guys, these intercalated discs are for quick transmission of impulse from one branch to another. Quick transmission of impulse. Please tell me these points are clear to everyone. Yes, please. Yes, guys, please tell me these points are clear to everyone. Ma'am, please teach English students as well. Nehal Dhingra. So far, I've always been teaching the English students. This is the first time I am catering to English students. So at that time, English students were saying, ma'am, please teach in English. Now bring that demand, teach in Punjabi. That is the only thing left. So I know Hindi, I know English, I know Punjabi. I've done it in 2 languages. Punjabi is left. Alright guys. Yeah, Dhanushri is right. It is a crossbreed of striated and non-striated, but don't say that in NEET. Okay. So are these points clear? Now talking about, see, flagellar movement, it has mentioned here. Where does flagellar movement, where is flagellar movement seen in the body? So it is seen in the spermatozoa. That is, your sperms have tail and they move in the flagellar form, and they maintain water current. Flagella move, they maintain water currents in the canal system of sponges. So basically they are talking about the collar cells or the conocytes. We have already read, they are flagellated cells. Collar cells or the conocytes, they maintain the water current and show flagellar movement. So protozoans in euglena, they also have 2 flagella, one is short, one is large. Muscles, now talking about the muscles, yes, come back. Let's talk about the muscle now. So guys, muscle is a specialized tissue which is a mesodermal origin. It constitutes 40 to 50% of the body, and it has these 4 properties, special properties guys, which is excitability, contractibility, extensibility and elasticity. So I think we have already discussed, I call it 3E plus C, right? So excitability, extensibility, elasticity and contractibility, and there are 3 types of muscles which I have just discussed. And see, I've already told you skeletal muscle is striated, so it is striped, so it is also known as striated. It is under your will, so voluntary. It is attached to your muscle, attached to your bones, hence it is also known as skeletal muscle. So the function is in locomotion and in changing the body posture. Then coming on to the visceral, where are they present? Inner walls of hollow visceral organs, your alimentary canal, your reproductive tract, your respiratory tract, all these structures, they have visceral muscles. Your blood vessels, they show constriction and dilation because of visceral muscles, right? They are smooth, they are non-striated, they are not under your control, hence involuntary. So they help in the transportation of food, transportation of gametes and so on. Is that clear? Then talking about the cardiac muscles, they show branching pattern. Where are they present? Nowhere else other than heart, and they are involuntary in nature, but appearance wise they are striated. They are involuntary and they are not under the control of activity of nervous system directly. We know that because our heart is auto excitable. Remember, it has its own structure, that is SAN, and that is why we call it myogenic also. So your heart can be influenced by nervous system, but your nervous system cannot generate the impulse. Please tell me that all these points are clear. Yes, guys. Please tell me all these points are clear. Yes or no? Right? Okay, guys. Now, talking about the detail of your skeletal muscle. See, your skeletal muscle, your muscle is made up of muscle bundles. Bundles are also known as fasciculus, right? Your bundles are made up of muscle fibers, and now all these bundles are held together by fascia. So see the hierarchy. Muscle has bundles, and bundles have muscle fibers. See the structure here. See the structure here. Yes, please. So see, this entire structure, this represents your muscle. This represents the muscle, right? Yes or no? Now, within the muscle, we have this as muscle fiber. These are muscle fibers. Do we see? Muscle is made up of muscle fiber, sorry, muscle bundles, not fibers. So the muscle is made up of muscle bundles, also known as fasciculus. Do we see these? These are the bundles. Yes. Now bundles are further made up of, yes, bundles are further made up of muscle fibers. Yes or no? So these are all fibers. These are all muscle fibers, and the fiber is equivalent to the cell. That is why we call it a muscle cell. So the terminology that we are going to use here is going to be entirely like that of cell. Is that clear? So the plasma membrane of fiber, the cytoplasm of fiber, although we will give them some separate names. See here. So this is the muscle fiber taken out. Is that clear? So this is the muscle fiber. Let's see the structure in detail now. Let's see the structure in detail. So I'm going to draw this before we read NCERT. Okay. Now see the muscle fiber. So this is the muscle fiber. We have just seen, I've told you how does the muscle, sorry. Okay. Okay, see here. So this is the muscle fiber. I've told you it is cylindrical in nature. It is multinucleated. Nucleus is towards the periphery, yes or no? And therefore it is called a syncytium. So the muscle, this is the plasma membrane, and plasma membrane is known as sarcolemma. It is known as sarcolemma. The cytoplasm here would be known as, cytoplasm will be known as sarcoplasm, right? The mitochondria will be known as sarcosome, yeah? And then this plasma membrane moves inward. This plasma membrane moves inward like this, and it forms these T-tubules. It forms these transverse tubules or T-tubules. Why? What is the significance? We'll understand. And in between the T-tubules, guys, is present the smooth endoplasmic reticulum. Smooth endoplasmic reticulum. We call this sarcoplasmic reticulum. Sarcoplasmic reticulum. It is a smooth endoplasmic reticulum. Remember that. It is SER. And it is a storehouse of calcium ions. It is a storehouse of calcium ions. Is that clear? Now, these three things, the two T-tubules and one SER, this is going to constitute a triad. Is that clear? These three things are going to form a triad. Is that clear? Now, this is where the message comes in. So, this is where your neuron, the motor neuron, gets the message. This is the motor neuron. The motor neuron is going to release the neurotransmitters here, right? So, this is acetylcholine. So, this junction between the motor neuron and sarcolemma, this constitutes your neuromuscular junction. So this is where neuromuscular junction is present. Neuromuscular junction. This is the synapse between the neuron and the muscle. And on your muscle, remember, on your muscle fiber, guys, sorry, on the sarcolemma of muscle fiber, we have these acetylcholine receptors. Remember this, because in the end of the chapter, we are going to discuss a disease related to these receptors. Please tell me these points are clear, right? So this is how it looks like. This is the structure of a muscle fiber. Now, within the muscle fiber, guys, within the muscle fiber, we have myofibrils. So within the muscle fiber, we have myofibrils, right? These are myofibrils. And myofibrils are further made up of myofilaments. They are further made up of myofilaments. So if I show you here, okay, never mind. So these are the muscle fibers, and now see, in between these there are going to be present some filaments like this. Okay, so these are the filaments that we will be discussing. So myofibrils will have myofilaments, and there are two types of filaments present in our skeletal muscle. Please tell me these points are clear. So here it mentions about muscle fiber, sarcoplasm, many nucleus, syncytium, sarcolemma, sarcoplasmic reticulum, reticulum, storehouse of calcium ions. Are we clear till here? Yes, and all these fibers, they are present parallelly arranged in the sarcoplasm. Within the fiber are present parallelly arranged are myofilaments. Have we seen it here? Parallelly arranged myofibrils. Please tell me. Or myofilaments. And each myofibril has alternate light and dark bands produced because of two important proteins coming onto this, actin and myosin. Please tell me these points are clear. Yes? Now guys, I am going to enhance this structure now. Now how are these structures present? So I am going to form, so there are two types of proteins present. So basically one is actin. These are the two major proteins, because other than these two also there are other proteins present. Two major proteins would be actin and myosin. Is that clear? This would be responsible for forming the light band along with other proteins. Also we call it I band or isotropic. I stands for isotropic. And then we have myosin is going to be a part of dark band, right? And this is also known as A band or anisotropic. Now, how they are going to collaborate to form these bands, let's discuss this. Okay, now going to talk about, okay, so see, let's say this is, make it thicker, okay, this is your thick band and so on, right? And then surrounding this we have, surrounding this we have these thin filaments. And this, is that clear? Like this. Now please tell me these points are clear. Okay, now see what you see here, right? Okay, now guys, what you see here, these red filaments are the thin filaments. Is that clear? The red filaments are the thin filaments and the blue filaments are the thick filaments. Do we understand this much? These are the thick filaments and these are the thin ones, right? Are made thick as thick. Now, these zigzag lines that I've drawn here are the Z lines. These are the Z lines. Okay. Now, this thin filament, yeah, that you can see here, this thin filament is actually made up of 3 different proteins. We are going to talk more about it. Okay. Now, thin, but they are so thin that light can still penetrate through, and hence it is going to form the light band. How? We'll see. Now, once more, I'm going to make a thick filament here. Yeah, I'm going to make a thick filament here, like this, and like this, and so on. Now watch. See, so this whole unit that I've made between the Z lines, this unit is known as sarcomere. This is one sarcomere, and this sarcomere is the unit of muscle contraction. So muscle contraction is going to take place here. Is it clear? This is the unit of muscle contraction. Are these points clear? Now, do you see that in between the two Z lines, there are these red lines which are thin, and then there are these thick lines, right? Now, let's talk about each filament one by one. Now, this thin filament, guys, this thin filament is made up of 3 different proteins. One is actin, then we have tropomyosin, and we also have troponin. These are the 3 proteins which are going to constitute your thin filament. Is that clear? While the thick filament is going to be formed of myosin. This is made up of myosin. Clear? Now, this thin filament is attached to this Z line, which is flexible. Okay, and the thick filament is also not held in air. It is also attached through another membrane which is present right in the center, and we call this membrane as M line. We call this as M line. Is that clear? Do you see that? Now, this area, see very carefully, this area, this area which extends till here. So as long as we have this thick filament, this entire area, this is thick, so light cannot pass through it. This is going to constitute your dark band or A band or anisotropic band. Yes? I'm going to ask you questions. Be very clear. Be very vigilant now. Right? Now see, so wherever there is a dark band, this thick filament, it is going to constitute your dark band. It is going to constitute your dark band, right? This is all dark band. This is another sarcomere. This is another sarcomere, but wherever we have this thick filament, it is going to constitute the dark band or the A band. Is that clear? Now, do we see the area in between? Area in between this band is the I band. This band is the I band or the light band. This band is the light band. Please tell me this point is clear, or I band or isotropic band. Please tell me these points are clear. Now, can I say, listen, can I say that dark band has both thick band and thin filaments and thick filaments? See, this is the region of the dark band. And on the outer area, do we see the thin filament is also getting into the dark band? Yes or no? So can I say that dark band contains both thick and thin filaments? It is made up of thick and thick filaments. Please tell me, yes or no? But unlike that, do we see that thin filaments, the I band is made up of only thin filaments? Yes or no? Also, do we see that Z line is this Z line is dividing the light band, because light band is half light band is on this side, half light band is on this side. So light band bisects the I band. Please tell me, can we say that? Yes or no? Now, just now I told you that the dark band is the position is that region till where the thick filament extends, and it comprises of thin filaments on the periphery and thick filament all throughout. Do you see that there is some space in the dark filament, in the dark band? There is this central region where there is no overlapping of thin filament. Yes or no? This region where there is no overlapping of the thin filament, this is known as, guys, H zone. So this is the central region of the dark band where the thin filament is not present. Can we say that? Yes or no? Can we say that? So H zone, thin, absent. Thin filaments as absent. Only thick. It only consists of thick filament. Please tell me these points are clear to you. And M line is present in between the H zone. Please tell me all these lines are, all these points are clear to you. Please tell me. Yes. Okay guys, now I want the response here. Give me a thumbs up. Let me know. Are these all points clear to you? Yes or no? Quick, quick, quick. Yes? 100%? Cool. Let's proceed then. Let's proceed then. Okay? Perfect. So then let's have a quick reading of what NCERT says. We have already seen this diagram, told you, and I've already discussed this, that there are two important proteins. See, they're writing two important proteins. It doesn't mean that only proteins. So it is actin and myosin. Light band contains actin and is called I band, while dark band contains called A band, or anisotropic band contains myosin. It is not writing myosin only, because they can't write myosin only, because we have seen that dark band has actin as well, thin as well as thick filament. So only will make the statement wrong. Is that clear? We can't write only here. Now see, so the actin filaments are thinner, yes, as compared to the myosin. Hence they are called thin and thick filaments. So we read in the center of the I band is the elastic Z line which bisects it. Filaments are firmly attached to the Z line. A band also held together in the middle by M line. A and I are arranged alternately. Functional unit. So the portion of myofibril between two successive Z lines is the functional unit known as sarcomere. Thin filaments on either side of the thick filament particularly overlap the free ends, leaving the central part of the thick filaments, and this central part of the thick filament not overlapped by thin filament is called the H zone. So I hope you can understand these lines now. This is NCERT. So they have shown this as a thin filament. This is a thin filament, and do we see that these dark red colors, these are the thick filaments, and as far as the the thick filament extends, this is the A band. Please tell me all these points are clear. And in the center, guys, in the center we have this H zone, the part of A band which is not overlapped by thin filament. This is called the H zone, and between the two Z lines, this is Z, this is Z, Z line. In between the Z line, we have two Z lines. We have a sarcomere, which is a functional unit. Is that clear? Yes or no? And now remember, whenever there will be the movement, certain changes are going to take place. What are the changes? Changes in sarcomere when the contraction occurs. Changes in the sarcomere during contraction. So let's learn it right here, because we already have read this. So what are the changes? What about the Z line? What about the I line? Yes, quickly let me know in the chat what happens to each part. Quickly guys, I band. Yes. Quickly let me know what happens to each. What happens to the H zone? All right. So what about the Z line? Comes closer. What about the I band? It reduces or smaller. A band remains the same, and H zone also reduces. So only one thing, one element that doesn't change, guys, that is nothing but your A band. It remains the same. I hope these points are clear to you all. I band decreases, Right. So rest, everything remains the same. Yes. Now let's go into the details of these thin filament and the thick filament. So talking about the thin filament, it says thin filament is made up of two filamentous actins. So guys, these are the two filament. This is one filament, and each F actin is made up of polymer of G. So this round thing that I'm drawing, this is the G actin. This is the G actin, right? Now remember, on this G actin, this is the G actin. G stands for globular. On this G actin, guys, we have myosin binding site. We have myosin binding site. Please tell me these points are clear. And now this will polymerize, this G actin will polymerize to form F actin. And how many F actins are there? Two actin. This is one F, and then there is another F actin. So this will be another F actin. Do we understand? Yes or no? Please tell me. These are the two filaments of F actin. Now we know that on F actin are present the myosin binding sites. Yes, so all these will have myosin binding sites. Now just imagine if these myosin binding sites are just exposed, what will happen? Myosin will quickly bind with it. And if your actin and myosin are always bound to each other, you will be in a constant state of contraction. Your body will be in a tetani permanently. So we can't let this happen. So what happens? We have, guys, we have this tape-like structure. Okay, now this has become quite thicker. Okay, so we have this another protein which covers these myosin binding sites like this and then like this, let's say. So this is hypothetical actually. It passes through the grooves, but I am just making it for your understanding. So it passes through these grooves. So this is tropomyosin. This is tropomyosin. And again, we have two filaments of tropomyosin. Please tell me these points are clear too. And on regular intervals here, at regular intervals, guys, we have this third protein known as tropomyosin. We have this tropomyosin, and this this white is tropomyosin. These red things that I've drawn, these are troponins. This is the troponin present at regular interval. Now see, this troponin has three subunits. One of them is known as TPM, another is known as TPC, and this one is known as TPI. So even if you skip that, we can go, we can do without it, but okay, this is TPI. There are three subunits. Now one of the subunits, especially this TPC, this is the subunit which C stands for calcium. It is going to bind with calcium. This TPM binds to tropomyosin. And I stands for inhibition. This is going to inhibit actin myosin from binding to each other. Inhibits actin and myosin to bind from each other. Please tell me these points are clear. Yes, guys, please tell me these points are clear. Yes or no? Right? So this is how your thin filament is produced. This is all about thin filament only. So your thin filament, as I told you, it is made up of three different proteins, which are actin, F actin, one. We have two filaments of troponin, this is the second, and we have three subunits containing the third protein, that is troponin, at regular intervals. Are these points clear? So this forms your thin filament. And then we have a thick filament which is made up of, again, a monomer known as meromyosin. So how does this meromyosin look like? It has this snake-like structure. It has this long tail. So from here to here, this is the tail, and then it has a short arm. This is the arm, and on top of it, it has a globular head. This is the head, and on the head, guys, it has two locations. One is the actin binding site. See, on actin we have myosin binding site, while on myosin we have actin binding site. So this is already a PYQ. There are two places on head One is actin binding site, and this is where ATP will bind. Now this indicates that contraction is an active process. It is going to require ATP binding site. These are both present on head region. Now remember, the tail will be known as light meromyosin, while the head and the arm, it is going to constitute heavy meromyosin. So this is what has been written, two parts. One is heavy meromyosin constituting tail and short arm, short arm and head, and tail constituting the heavy mero. So basically head and arm, these constitute your light meromyosin, and tail constituting your heavy meromyosin. Is this point clear? So head is an active ATPase activity. ATPase, because ATP will bind, ATP will break, so it has ATPase activity. ATPase is going to break down ATP. So ATP breakdown will take place. Hydrolysis in the presence of water. So it will form ADP plus IP. This is going to happen here. Is it clear? It has binding sites for ATP and for actin. Please tell me these points are clear to you all. And this is your NCERT version of the diagram. Do you see? Do you see this? So these green things, these are your F-actin. How many filaments? 2. Then we have this tropomyosin. What is the function of tropomyosin basically? So tropomyosin, it prevents, so it masks basically, it masks the myosin binding sites. It masks the myosin binding sites of actin. Please tell me these points are clear. And then we have troponin at regular intervals, and one of the units is going to bind with troponin. Is it clear? And this being your meromyosin, this is the meromyosin, not myosin, it is a myosin monomer. Do we see? This portion, this represents the tail, this represents the tail or the light meromyosin, and rest of it, which is called the cross arm or the heavy meromyosin. It has tail, head with these actin and ATP binding sites, and this is the arm. This is the arm. Please tell me these points are clear to you all. Yes, guys. Let's have a quick thumbs up break. Yes. Let's have a thick thumbs up break. Let me know. Quickly, guys. Let me know. All clear so far? All clear? Perfect. In paragraph, I have written tail as the HMM. Huh? Where did I write? No, the tail is H. Oh, sorry. I made this mistake. This is the wrong. Yes, tail, this forms heavy meromyosin and tail forms the light meromyosin. Yeah, you're right. Is it clear now? Clear? Okay, that was just by mistake. Thank you. Thank you, Siba. All clear? Session will be as long as it takes to complete the human physiology. So there is no upper end. It will go definitely till to 11 o'clock. Okay, so we'll keep taking 2-3 minutes break in between so that we don't get tired. We can have water breaks in between, but the session will go on. All right, guys, moving on to now mechanism. Now we have read the structure. We have read the structure of the proteins. So it's going to occur at the protein level, right? Now let's see how the mechanism occurs, how the muscle contracts. Now the best theory to explain the muscle contraction is a sliding filament theory. We have a sliding filament theory. Now as the name indicates, see, sliding filament, it says that the thin filaments slide over the thick filaments. It also means that it is a thin filament which is going to move. This moves, and the thick filament is static. Thick filament is not going to move. Thick filament is the one which is going to pull the thin filament onto it. Is that clear? Now let me use a little prop here. So see here, let's say this is the thick filament. Okay, so this is a thick filament, this is a thin filament. Thick filament is not going to move, but it is going to pull the thin filaments on the inner side. So there will be two thin filaments, right? So thin filaments are going to move in this way and this way. Are you getting that? So it is a thin filament that moves and the thick filaments remains static. So this is a very important line, guys, very important line. The thin filament moves over the thick filament. So please be careful what the NCERT writes for you. Ma'am, please send the previous classes. Lavanya, I have already sent this previous classes on my Telegram channel, and even Wasim sir has shared it on his channel. And as far as the part 2 notes are concerned, so part 2 and part 3 notes will be shared with you in the compiled way. So I'll compile the 2 notes because it is complete human physiology. So you will get them together. Is that clear? All right, guys. Now see what happens. So what is going to happen? So see, we have discussed this diagram. Let's go back to this diagram. Let's go back to this diagram. Yeah. So now talking about the mechanism of muscle contraction. So first thing first, the muscle, so the impulse comes through the motor neuron. Is that clear? At the neuromuscular junction. Nerve comes to the neuromuscular junction through motor neuron and it releases the neurotransmitter called acetylcholine. Acetylcholine is going to bind with these receptors and this means transmission of impulse to the inner regions. So as soon as the impulse reaches this muscle junction, this impulse is also transmitted to the sarcoplasmic reticulum. And that is the function of T-tubules, quick transmission of impulse to the SER. Now as soon as impulse reaches the SER, now this calcium starts coming out. So there is calcium efflux. There is calcium efflux. Please tell me these points are clear. So arrival of impulse at neuromuscular junction, binding of acetylcholine to the neurotransmitters, then transmission of impulse to SER, then release of calcium into the sarcoplasm. Please tell me if these points are clear to you all. Please tell me these points are clear to you all. Right? So we have discussed till here release of calcium ions into the sarcoplasm. Right? Now what will happen when this calcium binds to, now let's go back to this diagram. Now see, so I told you calcium is going to bind onto troponin. This is where calcium is going to bind. Calcium binds one of the troponin. Remember TPC I told you. So when calcium binds to TPC, this tropomyosin, which is masking the myosin binding sites, this will move. Okay? When TPC binds to, calcium binds to TPC, tropomyosin will move and it will unmask the myosin binding site now. And when this will be unmasked, so the myosin binding sites here, so F-actin has myosin binding sites. So these myosin binding sites will be exposed. Please tell me these points are clear. Do we understand till here? Right? Now, simultaneously, what's happening? Simultaneously, this ATP binding site, simultaneously ATP on the ATP binding site is getting hydrolyzed, means breaking down in the presence of water, hydrolyzing into form ADP and phosphate. And what type of activity is occurring here? ATPase. Yes or no? It says it has ATPase activity. So this ATPase enzyme is going to break down ATP, and what will happen? As soon as this energy is released, ATP breakdown occurs and energy is released, this head is going to bind to the myosin binding site. So this actin binding site and myosin binding site, they will form a junction. They will cross bridge. They will form a cross bridge. They form cross bridge. Please tell me these points are clear. What did I say? Message has reached. Calcium has been released in the sarcoplasm. Calcium binds to TPC. this tape moves away, it unmasks the myosin binding sites, and simultaneously ATP breakdown takes place, and now this actin binding site binds with the myosin binding site of actin. So it's like this. Let's say this is the myosin and this is the, sorry, this is the actin and this is the myosin head, right? Now this head is quickly going to bind like this. And once it is bound, it is going to move backward. And when it will move backward, it will pull the thin filament inward. So this is how the thin filaments are going to slide over the thick filaments. Do you get that? Please tell me these points are clear. Please tell me these points are clear. So see here, quickly, it's signal sent by the central nervous system via motor neuron. Motor unit is formed. Junction is, so the junction between sarcolemma is known as neuromuscular junction or motor end plate. So a neurotransmitter is released in the form of ACH, acetylcholine, generates the action potential in the sarcolemma. What will happen? Calcium ions released in the sarcoplasm, calcium binding to the subunit of troponin. So that subunit is TPC, right? So removes the masking of actin sites for myosin, means unmasks. Please tell me these points are clear. And then utilizing the energy from ATP hydrolysis, myosin head now binds to the exposed active sites on actin, forming a cross bridge. Please tell me that these points are clear to you all. Yes, guys. Please tell me these points are clear to you all. Yes, guys. Quick. Please tell me these points are clear to you all. Mic is already near my mouth. Quick. Please tell me these points are clear. Okay. Right? All right then. Now, see, this is the diagrammatic representation of what has been written otherwise. So it starts from here. Let's say, so basically this is actin filament, this is a myosin filament, this is the head. So ATP breakdown is taking place. So where ATP hydrolysis occurs, and then myosin binds to the actin and cross bridge is formed. See, cross bridge formation, actin and myosin binds, cross bridge formation. And when the head moves backward, moves backward, told you, moves backward. So that results in sliding or rotation. So thin filament, see, remember, it binds, cross bridge form, and then head moves backward. When it moves backward, it is going to slide it like this. And then finally, it has to relax also. So then ATP regeneration will occur. So when ATP comes back on regeneration, this is going to break the cross bridge. When the cross bridge is broken, it is relaxed again. It is relaxed again. Please tell me all these points are clear to everyone, right? So already told you about these things. So these are the changes which are going to occur during the muscle contraction. A band retains the length. There is only one structure which will not change, that is the A band. Okay, and then when new ATP binds, then the cross bridge is broken and it goes in the relaxed state. So this is the structure, filament theory, sliding filament theory of muscle contraction, showing the relative size. Do we see? This is the original structure, and this is the muscle fiber in relaxed state. This is contracting. That is why Z lines have come closer. So Z lines were here, they have come closer, right? And more contraction, so they will come even closer. So earlier the H zone was this big, and now H zone has become shortened, and on maximum contraction we see there is no H zone at all. No H zone. It is in maximum contraction. Please tell me all these diagrams are also clear to you all. And we have discussed this. So what will happen during relaxation? So first of all, ATP regeneration. Also, message has to stop coming. So no impulse. So basically calcium will go back. So calcium influx into SER. And then myosin binding sites will be masked again. Myosin binding sites are masked again. Please tell me these points are clear to everyone. And now very important line, that repeated contractions, guys, will lead to lactic acid formation, anaerobic breakdown of glycogen, which is known as muscle fatigue, and this occurs because of myoglobin. Now see, there are two types of muscle fibers actually. One is the red fibers, red muscle fibers. Okay, so how quickly your muscle will show fatigue, it depends on this pigment called myoglobin, which is red in color, and it is an oxygen storing pigment. Please note down, hemoglobin is oxygen carrying or transporting pigment, while this holds to oxygen and it is oxygen storing pigment. So there are two types of red fibers, guys, red muscle fibers and white muscle fibers. White muscle fibers. I am talking about your skeletal muscle fibers only. The skeletal muscle fibers are of two types. Okay, skeletal muscle fibers are of two types. One is red, another is yellow. Another is white. Red and white. Now, what makes it red? So basically, red is red because of more myoglobin. So it has more myoglobin. So myoglobin means more oxygen. So more oxygen. If it has more oxygen, it will have more mitochondria so that it can produce more energy, right? But so they don't undergo fatigue very quickly. So do not fatigue easily. Do not fatigue easily. So they can work for a longer period of time. But more oxygen and more mitochondria also means they will undergo slow contraction. And for the slow contraction, they will show less SER and less calcium. Is it clear? The opposite is true for white. It is white because less of myoglobin. Less myoglobin means less oxygen. Less oxygen means it will also have less mitochondria. It means it will undergo fatigue. They fatigue easily. You will get tired quickly. Fatigue easily. Why fatigue easily? Because they show contraction, quick contraction. They show quick contraction, and for quick contraction, guys, they have more SER and more calcium ions. It is opposite. Do you see the difference? Please tell me, do you see the difference here? Yes, guys, do we see the difference here? Yes or no? Thank you, Senthil. All right, guys. So that is why, because they don't fatigue easily, we call these muscles as aerobic muscles. So another name for red muscle fibers, aerobic muscles, and these ones will be known as anaerobic. Is it clear? Rest is all written and we can read. Pale, whitish, and so we call them white. Please tell me these points are clear to all. And now coming to the second part of this chapter, for movement and locomotion, the second thing that we need is the bones. So let's start with the skeletal system. It comprises of, it comprises of bones and few cartilages, right? So all these activities, now both of them, they are basically specialized connective tissue. So bone has hard matrix due to calcium salts and later is slightly pliable due to chondrin salts. And how many bones do we have? 206 in adult and few cartilages. And these 206 bones are divided into two groups. Let's talk about them. So the two groups are axial skeleton and appendicular skeleton. In axial skeleton, we have 80 bones which are differentiated into different regions. Let's understand. So axial skeleton, guys, axial skeleton has axial skeleton has, so I'm going to make a flow chart here itself. Axial skeleton has skull, it has a hyoid bone, it has a vertebral column. So NTA has already clarified hyoid is not a part of skull, vertebral column, and it has sternum and it has ribs, right? So these are the components of your axial skeleton, right? Now see, how many bones are present in ribs? So ribs are in total 12 pairs or 24. We have one sternum, we have 12, sorry, we have 26 bones in the vertebral column called vertebrae. We have one hyoid, and then skull is further differentiated into cranial bones, cranium, that is forming your brain box, which has 8 bones, and it also forms 14 facial bones. So in total, there are 22 bones in your skull. Are these components clear? Please tell me. Is this classification clear to all? Is this classification clear to all? Yes? Hyoid is not a part of skull. This has been clearly mentioned now. Present at the base of buccal cavity. Yes? Now, let's discuss, just a second. So we have 22, 23, 24, 24 and 24, 48, 48 and 26. We have this. Please tell me these points are clear. Yes, guys. And also we have, yes. Please tell me these points are clear. Yes? Yes, please. Anyone with any doubt? Vomer is unpaired. Vomer is unpaired. Yes, guys. Please let me know. Let me see your chat. What are you writing here? Yes, guys, quickly. Please let me know. All these points are clear to everyone. Right? So these are 74 bones, and we need one more dabba. And we have ear ossicles. Is it clear? How many? We have 3 pairs or 6 bones. All clear? Now these are the smallest bones of your body. Clear? All right, now discussing them one by one. So see, facial region, this is this. Let's discuss this point. So a single U-shaped bone called hyoid is present at the base of the buccal cavity, and it is also included in skull. Now guys, please note down, those of you who are using the old book, this is not included in the skull. It is clearly written, it is not a part of skull. Please note down, it is not a part of skull. And then in the middle here, guys, we have 3 tiny bones, malleus incuscatus, collectively called ear ossicles. Skull region will articulate with this, and we have a dicondylic skull. Now before we go on to the dicondylic skull, I want to discuss the details of cranial bones. Now see here, now which are the cranial bones? So which are forming the cranial bones which form the brain box, right? So guys, in the front we have this frontal bone. We have a frontal. We have one frontal. We have 2 parietals, right? We have 2 temporals. Then, and how many are there? Yes, we have one occipital. We have one sphenoid. And we have one ethmoid. So how many? 3, 4, 5, 6, 7 and 8. So these are the cranial bones. So you should know which of them are paired. it is only these two bones which are paired, rest all are unpaired. Clear? Now talking about your facial bones. Facial bones are 14 in number, right? So which all are the facial bones? So first of all, your nasal bones here. The nasals, 2. Then to side, we have cheekbones called zygomatics. Again, these are the cheekbones forming 2. Then we have 2 maxillae, right? And we have 1 mandible, the lower jaw. We have 1 vomer. We have 2 lacrimals. Yes, please. What else? We have 2 palatines. Please tell me all these points are clear. So how many are these? 1, 2, 3, 4, 5. That is 10, 11, 12. And what else? Yes. Which are the other 2 bones here? Which are the other 2 bones here, guys? Quickly let me know. Quick. Let me know which are the other facial bones. Yes, please. So we have 14 bones. Which are the other 2 bones present here? Yes, please. Vomer is done, maxilla, lacrimals, palatine, and then there are 2 which are not really mentioned. Yes, nasal conchae, right? So actually we have 3 conchae, but the bones are 2, so they are nasal conchae. Conchae bones, these are 2. So this makes us 14 facial bones. Please tell me. So you should be thorough with the labeling as well. Now, moving on to the vertebral column. Your vertebral column, guys, is constituted of 26 bones arranged. And where are they present? They are dorsally placed at the base, starting from the base of the skull, and it constitutes the main framework of the trunk, right? So now like this. So all these 26 bones, they are different. They are differentiated into 5 regions, and we have this formula to remember that. So we have 7 vertebra in the cervical region. We have 12 in the thoracic region. We have 5 in the lumbar region. We have 5 fused to form 1 in the sacral region, and we have 4 fused to form 1, that is in the tail region or the coccygeal region. So how many in total? We have 26. If we talk about the initial stage, that is the early stage of our life, then these will not be fused and they'll count to 33. Is that clear? Now they are saying that each vertebra has a central hollow portion that is called the neural canal. So basically it's like this. Let's say if this is one of the vertebra, so we have this hole in between, and through this hole, guys, basically your spinal cord will pass. So this hollow space is known as the neural canal, through which the spinal cord passes. Now the first vertebra is known as atlas and articulates with the occipital condyles. So it is in the occipital bone, and second one is the axis. So they have already written all these things which I have compiled in the form of a formula, right? Now, most of the mammals, see, there are 7. The number of cervical vertebra are 7 in almost all mammals, not 100%, but almost all. And what is the function? It protects the spinal cord, it supports the head and point of attachment for the ribs, and also the musculature of the back. Is this point clear? Now on the dorsal side we have vertebral column, while on the ventral side, guys, we have sternum in the ventral midline of the thorax. This sternum is a flat bone. Are these points clear? Right? So this is the structure shown. So this is the cervical, this is the thoracic, this is lumbar, sacrum and coccygeal, and in between the two, guys, this is the intervertebral disc. Now the question here is, intervertebral disc is made up of, please tell me, it is made up of what? It is made up of cartilage. It is made up of cartilage, fibrous cartilage. Is that clear? It is made up of fibrous cartilage, which is also known as disc. Please tell me these points are clear. Right? Now moving on to the next. So talking about the ribs now. See here. So there are 12 pairs of ribs. Sorry. There are 12 pairs of ribs. Now each rib is a thin flat bone connected, it is connected dorsally. On the back side, it is connected to the vertebral column and ventrally to the sternum. So that is why we have bicephalic. Our ribs are known as bicephalic. This time also we got a question. So we have bicephalic because there are two articulation surfaces, one on the dorsal side, other on the ventral side. Now see, another point, the first 7 vertebra, the first 7 pairs, these are called the true ribs. Why? Because they are dorsally attached. They are attached dorsally to the vertebral column, and in front they are attached directly to the sternum. See, they are attached to the thoracic vertebra and ventrally connected to the sternum with the help of which cartilage? Hyaline cartilage. Then guys, we have 8th, 9th and 10th pair. They articulate directly with the sternum. Sorry, they do not articulate directly to the sternum, but join the 7th rib with the help of, again, hyaline cartilage. Is that clear? So we don't call them true ribs, we call them false ribs or vertebrochondral, while these are known as vertebrosternal. As the name itself says, dorsally attached to the vertebral column, ventrally attached to the sternum. While these ones, they are dorsally attached to the vertebral column, but ventrally they are attached to the hyaline cartilage and then to the 7th rib. So chondra word is for cartilage. Are these points clear? And then guys, we have last 2 pairs, that is 11th and 12th. These are not connected ventrally at all, and therefore these are known as floating ribs. We also call these floating ribs as vertebral ribs. We also call them vertebral ribs because they are only connected to the vertebral column. Is that clear? So thoracic vertebrae, ribs and sternum together constitute the rib cage. Important line. See, it is only the thoracic vertebra and the ribs and the sternum that will constitute the rib cage. So not all the ribs, but only the thoracic ribs. Are these points clear? So this is the structure. These are the true ribs. 7th, 8th, 9th are, and then these 2 are the floating. This is the dorsal vertebral column. These are the ribs. This is the sternum. This one, right? This one being the sternum, flat rib. Please tell me all these points are clear. And in between these, we see a disc, which is intervertebral disc, right, made up of cartilage. And this cartilage is not hyaline, it is white fibrous cartilage, very strong, right? So with this we complete our axial skeleton. And now moving on to the appendicular skeleton, guys. So appendicular skeleton, as the name says, these constitute the bones of the limbs and that of the girdles, right? So how many in total? So how many bones are present here? So 206 minus 80, that would be 126 bones. It constitutes 126 bones. So now see, there are going to be 30 bones in each limb. So in total 120 bones in total limbs and 6 bones present in your girdles. Is that clear? Now the bones of the hand. So what are the bones in the hand? That is a forelimb they are talking about. Humerus, radius, ulna, carpals, metacarpals, metaphalanges and phalanges. So metacarpals and phalanges. So these are the bones of your forelimb. These are the bones of your forelimb, right? While these bones, so it is femur, that is a thigh bone, the longest bone, the thickest bone of the thigh, tibia, fibula, tarsals, metatarsals, your knee, right, and phalanges. They in total constitute your hind limb, bones of the hind limbs. Now each bone, each bone will have, each limb will have 30 bones. So in total, so that's how we have 120 bones in the limbs. Are these points clear? And also this patella, this is also a part of, or ventrally present kneecap, this is also a part of your hind limb. Is that clear? So they constitute your hind limb. Please tell me all these points are clear. Please tell me all these points are clear. Right? Now talking about the girdles. Now girdles in total, there are two types of girdles, pectoral girdle and the pelvic girdle. So see, pectoral girdle is your shoulder girdle and this is the hip girdle, right? Now basically, why are these girdles important? Girdles help in the articulation of the upper and lower limb with the axial skeleton. This is the function of the girdles. Is that clear? Now each girdle has two halves. Each girdle has two halves, right? Now talking about the pectoral girdle first. Pectoral girdle, that is the shoulder girdle. It has one clavicle and one scapula. It has one scapula. Let's draw it here and see. See, so this structure, this is the scapula and this structure represents the clavicle. So clavicle and scapula, these two structures form your one side of the shoulder girdle or the pectoral girdle. Is that clear? Now it says scapula is large triangular flat bone. Can we say? Can we see? It is large, it is triangular, it is flat, right? And situated on the back side. So this bone that you feel is the scapula on the dorsal side between the second and the seventh rib. Where is it present? Between the second and seventh rib. So you should write all these things along with the diagram. So the dorsal flat triangular body of the, so how is it? Dorsal flat triangular body has slightly elevated ridge known as spine, which projects as a flat expanded process called acromion process. Now it may not be visible here. I'll draw how it is visible from the dorsal side. So basically, this is how your scapula is present. And on the dorsal side, guys, it has this ridge. This is called spine. And it says elevated ridge called spine, which projects as a flat expanded process. Now this process of the spine is called the acromion process. Are you getting that? Now what is the function of acromion process? The clavicle, this clavicle we have drawn here, the clavicle is going to articulate with this. So clavicle, this is clavicle. So clavicle is going to bind with the acromion process. Please tell me these points are clear. So this is the acromion process and this is the clavicle, that is the shoulder blade or the beauty bone which binds to it. Is that clear? So binds, this is the dorsal side. Do you see? This is the dorsal side. On the ventral side, it is going to bind to the sternum. This is on the so this is on the ventral side. So your clavicle is articulated to acromion process of scapula on the dorsal side, and ventral side it is articulated with the sternum. Are these points clear? So clavicle articulates with this. Below the acromion process is a cavity known as the glenoid cavity. Now see here, this cavity below this cavity is known as glenoid cavity, and it is to this cavity that the head of humerus is going to fit. So head of the humerus is going to fit here like this. This is the head of humerus. Do you see all these points? Please tell me these points are clear. Yes guys, please tell me these points are clear. Yes or no? So it articulates with glenoid cavity, articulates with the head of the humerus to form the shoulder blade, and each clavicle is a long slender bone with two curvatures. It has two curvatures again, and commonly known as the collar bone. Why two curvatures? Because one curvature is going to bind here and one curvature is going to bind to here. Are these points clear? Please tell me that this whole shoulder blade thing is clear to you. And also we can see the bones, humerus bone, this is radius, this is one ulna, then there are 5 bones in the carpals, sorry, 8 bones in the carpals, 5 bones in the metacarpals, and 14 bones in the phalanges. Are these points clear? This is in each upper limb. Now similarly, we have the lower limb, guys. Now the pelvic girdle, which is going to form your hip girdle, this is going to form your hip girdle. This consists of 2 coxal bones. Now see here, there are just 2 coxal bones. While in the shoulder blade, there are 2 scapula and 2 clavicles. So 4 bones, but here in the hip region, we just have 2 coxal bones, which are shown here. Do you see this? This is the coxal bone. This is one coxal bone. Is that clear? Now each coxal bone is formed by the fusion of these 3 bones, ilium, ischium and pubis. Is this clear? So see, it is written, it is formed by the fusion of 3 bones, ilium, ischium and pubis. Now the point where these bones fuse, that point, now that point where these bones fuse, this point is known as, point of fusion is known as acetabulum. And now this is where the thigh bone is going to articulate. So do we see the thigh bone? This is the thigh bone articulating here. That is the femur. Do you see that? Femur is articulating with the acetabulum. Like your humerus articulates with glenoid cavity, this fuses with the acetabulum. Now 2 halves of the pelvic girdle, they meet ventrally. So here, let's say here, there is another coxal bone like this, right? There will be another coxal bone. They're going to fuse ventrally here, constituting, here, they will constitute, see, they will constitute pubic symphysis, made up of fibrous cartilage. Again, this is pubic symphysis. So it is during the childbirth that this pubic symphysis, sorry, is going to open, widen. So just like your vertebral disc, that is intervertebral disc, your pubic symphysis is also made up of fibrous cartilage. Please tell me these points are clear. Also made up of white fibrous cartilage. Are these points clear? Yes, please. Are these points clear to all? Can we move on? Now this is a very important topic in the last topic of the chapter, that is joints. So joints is anywhere where 2 bones meet. It is a point of contact between 2 bones. Any 2 bones, right? Now see, or between the 2 bones and the cartilage. So wherever bone meets or bone cartilage meets, it forms a joint. Now see, there are 3 types of joints basically. One is the fibrous joint, 3 major structural forms of joints: fibrous, cartilaginous, and we have synovial joint. Now let's talk about them one by one. Fibrous joints are also known as immovable joints. They do not allow any movement. Is that clear? So we also call them immovable joints. They are totally immovable. What are the examples? Where are they present? They are present in the skull bones. We call these as sutures. So the joints in the between the skull bones are known as sutures. Is that clear? Then we have cartilaginous joints. These cartilaginous joints are partially movable joints. These are partially movable joints. Is it clear? Now, why are these totally immovable and these are partially movable? Because they are made up of cartilage, while these are immovable because they are made up of dense fibrous. They are made up of dense fibrous connective tissue, which is hard, which is difficult to move, while this still allows some movement. Is that clear? So these are found in between the adjacent vertebra, right? And it permits limited movement. So between the 2 vertebrae, when we move backwards, that is intervertebral disc, right? That is where this joint is present. Now coming on to the synovial joints. Synovial joints, they have a fluid known as synovial between the 2 surfaces, and that is why it allows considerable movement. It allows considerable movement. Now, very important topic. What are the different types of movements? Sorry, joints, synovial joints. We have ball and socket joint, right? We have hinge joint. Now, these can easily come in match the following. These pivot joint, gliding joint, we have saddle joint, and you must know the examples. See, ball and socket joint allows free movement. So we are able to rotate because there is this ball, which is the glenoid which is the head of the humerus, and this cavity socket is the glenoid cavity, and it can easily rotate, right? So it is between humerus and pectoral girdle, and also it is present. Second example is your femur and the pelvic girdle, basically acetabulum. Is that clear? Then hinge joint is a knee joint, as well as we have elbow joint. Both are the examples of hinge joint. Your pivot joint is between atlas and axis, which allows the movement of head, right? And then gliding joint is between the carpals as well as between the tarsals, that is your ankle joint or your wrist joint. So gliding is ankle joint between the tarsals and wrist joint between your carpals, wrist joint. Is that clear? And then we have saddle joint, very important, between carpal and metacarpal of the thumb. Please tell me all these points are clear to you. All these examples are super important. Yes, guys. And then we have these disorders, myasthenia gravis, very important, muscular dystrophy and tetani. All these are disorders related to your muscular system. See, myasthenia gravis is an autoimmune disorder where your neuromuscular junction leading to fatigue and this. What happens basically in autoimmune disorder? Your acetylcholine receptors are damaged. Antibodies destroy acetylcholine receptors. Is that clear? So no receiving of the message means your muscle will weaken, it will undergo paralysis. Is that clear? Then talking about the muscular dystrophy, guys, this is a genetic disorder where there is progressive degeneration of the skeletal muscle. And then we have tetani. In tetani, there is a rapid spasm. So muscle undergoes wild contraction, suddenly contraction, and due to low calcium level in the body fluid. Is that clear? Please tell me. And then talking about the disorders of bones, we have arthritis. Arthro word is for joint. Itis means inflammation. So this is arthritis. Osteoporosis, bones become porous. So age related, decreased bone mass, increased chances of fractures. This is due to decreased level of estrogen, because after decreased level of estrogen, calcium level decreases. Estrogen also helps in calcium absorption. And then gout, it is an inflammation of joints due to accumulation of uric acid. So we also call it gouty arthritis. Also call it gouty arthritis. Please tell me all these points are clear to everyone. You don't understand pubic symphysis. See, pubic region is where the two coxal bones meet, right? So pubic symphysis, symph word is for fusion, where the two coxal bones fuse, that is pubic symphysis. Please let me know, guys, that these points are clear. Let's have a quick break. Yes, guys, yes. Thumbs up break, water break, and then we are going to shift on to the next chapter, nervous system. Not a big chapter because NTA has removed most of the topic. Jyoti, what do you want me to repeat in disorders? Please be specific. Please be specific. Condylar joint is not given here, but yes, of course, condylar joints are present. They are also type of your synovial joint. Jyoti, you have to be very specific. What do you want me to repeat in disorders? Can't spam like this, guys. You have to be specific. All right, moving on to the next chapter, that is nervous system. So see, Nervous, now the last two chapters that we are left with, that is nervous and the chemical control and coordination, both these systems are involved in maintaining control and coordination, right? They maintain control and coordination. What is phosphogen that helps in the regeneration? So phosphogen, guys, is proteins. So it is a source of phosphate. Actually, phosphogen is a name given to phosphocreatinine. Okay, phosphocreatinine is present in your muscles. So Vishwa, phosphocreatinine is called phosphogen because it can donate phosphate for the regeneration of ATP. Okay. Yeah. All right, guys. Moving on. So coordination between two organs when they interact and all this requires two systems. One is your nervous system and second is your endocrine system. So these two systems are going to help in coordination. So see, it's writing the functions of muscles, lungs, all these are coordinated while performing physical activities. This is because your neural system and endocrine system jointly coordinate and integrate all the activities of the organs in the synchronized manner. So we'll be talking about their synchronization and control one by one. Now see, talking about the neural system. So how is neural control different from the endocrine role? So neural system basically provides point-to-point connection and there is quick coordination because of this. While endocrine system works through hormones and this system is slow. See, this system is slow, but the effect can be long lasting and it works through hormones, right? While this neural system is quick and short-lived, the effect is short-lived and works through nerves and neurotransmitters. Is that clear? Overall, neuron sends signal to an overall organ, while hormones are going to work at the cellular level. This works at the organ level, while this works at the cellular level. Is that clear? So it is written through synapse and all these things. Now let's talk about the neural system. So first of all, every system has a unit, right? We have talked about alveoli and we have talked about nephrons. We have talked about muscle fibers. Now going to talk about the unit of neural system, which is neurons, which can detect, which can receive, which can transmit different types of stimuli. Now these neurons, we have always been drawing a very elaborate structure of neuron, but it has not always been that elaborate. The neural organization is very simple in lower invertebrates. For example, in Hydra, it is just composed of a network of neurons, right? But gradually, in higher organisms, it was better organized in insects where a brain is present along with the ganglia and nervous tissue. And in vertebrates, we have even very well developed neural system. Now moving on to the next part. See, this is important. So overall human neural system is divided into two parts. One is the central nervous system and then we have the peripheral nervous system. So we know that central nervous system is comprised of your brain and spinal cord, and your peripheral nervous system comprises of all the nerves. Is that clear? So this is your central nervous system. This is your central nervous system, while peripheral nervous system consists of all the nerves around your brain and spinal cord. So there are two types of nerves, guys. So peripheral nervous system has two types of nerves. One is afferent and second is efferent. So afferent is which goes towards the central nervous system. See, afferent impulses from tissue to the central nervous system, while efferent transmit impulses from central nervous system to the peripheral nervous system. Is that clear? It's easy. So this is your central nervous system, spinal cord and brain, right? And let's say this is the organ. So the one which will carry message towards this is afferent. And the one which will bring message back is called efferent nerve fibers. Are these points clear? Right? Now peripheral nervous system will still have two divisions. One is somatic and another is autonomic nervous system. Why 2? Because somatic relays impulses to the central nervous system to the skeletal muscles, while autonomic is from central nervous system to involuntary organs. So this comprises, this is your SNS and this constitutes your ANS. Are these points clear? Are these points clear? So see, let's say if this is your skeletal muscle, and then there is another involuntary organ. This is your, or this is voluntary and this is involuntary. Okay. Now see here. Let's say this carries message afferent, this brings message, this is efferent. Now what is it saying? So if it is between CNS and voluntary, this is your somatic nervous system, right? These nerve fibers, afferent, efferent, between skeleton and this constitute somatic nervous system, and these ones between involuntary and central nervous system, these will constitute autonomic nervous system. Is that clear? And yet autonomic nervous system is of two types: parasympathetic autonervous system, and then we have sympathetic autonomic nervous system. This operates during normal situation and this operates during emergency. Are these points clear to all? And then we have visceral nervous system. It is a part of basically peripheral nervous system, comprises of whole complex of nerves, fibers, ganglia and plexus, traveling from central nervous system to the viscera and viscera to the CNS. Viscera means internal organs. We have a specialization for that. Please tell me these points are clear. Yes, please. Please tell me these points are clear. All of you. We don't need to learn the types of pseudopodia like philopodia. No, Shoaib, not at all. Don't waste your time, please. Don't waste your time at all. Okay, now talking about the neuron. Let's see the neuron. So this is the structure of, so there are three main parts. So one is dendrites. We have cell body, also known as cyton, also known as perikaryon. And we have the third part, that is axon, right? So what are the functions? This receives the stimulus. This generates the impulse. And your axon is going to conduct and transmit the impulse. Conduct and transmits the impulse to the next neuron. Is that clear? So these are the three parts. So there are structures written out here called Nissel's granules. What are they? Let's discuss. By the way, Nissel's granules are present in dendrites and cell body, not in here. So what is the structure like? Okay. Now these are the dendrites. Do we see here? These are the dendrites. This whole structure, rest of it, this whole structure, this is called cell body, and then rest of the part is called axon. Okay. Now Nissel's granules are present in dendrites as well as in the cell body, and what do? They are basically your endoplasmic reticulum and ribosomes, and they help in protein synthesis, neurotransmitters. This is the nucleus and this is the Schwann cell. This Schwann cell is going to produce the fatty layer called myelin sheath. Now there are spaces where myelin sheath is absent. That place is called node of Renvier. Now see, at node of Renvier, myelin sheath is absent, but Schwann cell will still be present. This is Schwann cell, this is myelin sheath. So Schwann cell is present irrespective whether myelin sheath is present or not. Is that clear? And then we have axon terminals and the synaptic knobs, and in synaptic knobs we have neurotransmitters. Please tell me these points are clear. Is that clear to everyone? So this is all about Nissel's granules. They contain Nissel granules. They're called dendrites. Fibers transmit impulse towards the cell body. Axon is a long distal end which is branched, and now each branch has terminated into a bulb-like structure called synaptic knob, which has synaptic vesicles. And synaptic vesicles contain these neurotransmitters. So axons are going to transmit the nerve impulse away from the cell body. Is it clear? to a synapse or to the neuromuscular junction. Now, depending upon the number of axons and dendrites, there are different types of neurons. See here. So, there are different types of neurons. So, let's say if there is no process at all, we call it apolar. So, coelantrates or like Hydra, they had such type of neuron. There is no process, it is just a cell body. Then we have unipolar, whereas there is just one process, and this one process will act like a axon, while this one is a cell body. This is called unipolar. So during our embryonic development, we have such type of neurons. Then there are two processes. When there are just two processes, this being the cell body, one process acts like a dendrite, other acts as an axon. This is called bipolar. So this is in the early embryo, while the bipolars are found either in retina or in the olfactory lobe, olfactory epithelium. Is it clear? And then we have this typical, this is the multipolar neuron. So multipolar neuron has many dendrites and usually one axon. So in most of our body, we have multipolar neurons. Are these points clear? So see, we have discussed about already bipolar, unipolar, and we have talked about the multipolar as well. They have mentioned unipolar in the embryonic stage, bipolar in the retina. I have already given you other examples as well. Now on the basis of, myelin sheath, there are myelinated neurons and then there are non-myelinated. So myelinated are enveloped with Schwann cells which form a myelin sheath, and the gaps between the two are called nodes of Ranvier. They are, now see, very important line, where are these present? Myelinateds are found in the spinal cord and cranial nerves, while unmyelinated or non-myelinated nerves, they are also enclosed by Schwann cell, but that does not form a myelin sheath. They are commonly found in autonomic nervous system and somatic nervous system. Please tell me these points are clear. Now here you can directly get the match the following, right? Now talking about the generation and conduction of nerve impulse. So for explaining this, I would use my own diagrams. Let's understand the nerve impulse here. Let's understand. Yeah. Now guys, let's say this is your nerve fiber. Wait. Let's say this is your nerve fiber. Okay. All right. Now when the fiber, your nerve fiber is at rest, so we call this resting membrane. Resting membrane means there is no impulse. No impulse yet. Right? When there is no impulse, now see, when there is no impulse, look at it carefully. When there is no impulse, your resting membrane has two poles. So it is negative on the inner side and it is positive on the outer side. Is that clear? How negative on the inner side? The potential is minus 70 millivolt. This is called a resting membrane potential. So this is how your membrane is present. So there are two poles developed here. So that is why the resting membrane is also known as polarized membrane. Is that clear? What else contributes to the polarization? Why is it negative on the inner side? Because there are negatively charged proteins here in the exoplasm. See, what are these structures, by the way, what you are writing? So this is the exolemma, right? And this is the exoplasm. And on the outer side, we have this extracellular fluid. Exoplasm can be known as intracellular fluid. Can we say that? So in the interstitial fluid, or intracellular fluid or exoplasm, we have negatively charged proteins, one. Also, there are less sodium and more of potassium ions in here. So this is how your exoplasm is present. Is that clear? While in the ECF, now see, in the ECF, that is the fluid outside the neuron, outside the axon, basically we have more of sodium as compared to potassium. Are these points clear? Now this membrane that we are discussing here, this membrane has a sodium potassium pump. It has a sodium potassium pump. Now this membrane is continuously pumping out. See, there is more sodium on the outer side. So sodium is continuously coming in. See, there are some channels present here. There are some channels coming here. So since there is more potassium on the inner side, so potassium is going out. Listen, so potassium is going out, and through such leaky channels, sodium is also coming in. Potassium going out. This is normal diffusion. Why? More sodium on the outside as compared to inside, and more potassium on the inside as compared to outside. So what is happening? The sodium potassium pump is maintaining that there should be more potassium on the inner side. So it is pumping in potassium, it is pumping out sodium. 3 sodium out and 2 potassium in at the expense of 1 ATP. This is what your sodium potassium pump is doing. Please understand. Please understand this. Is that clear? So your membrane is negatively charged because there are negatively charged proteins, and then there are more potassium ions on the inner side as compared to the outer side. Outer side has more of sodium. Now there are certain channels which are always open. So through these, sodium will pump in, potassium will keep pumping out, but we need to maintain this situation. So for that, sodium potassium pump is continuously working. At expense of 1 ATP, 3 sodium goes out and 2 potassium taken in. Now remember, we say that this resting membrane or polarized membrane is more permeable to potassium ions. Please remember. Now, what's next? The next thing is, as soon as the stimulus arrives, so there is arrival of stimulus. What will happen? So basically, this is going to change the polarity. What will happen? The polarity is going to change. So as soon as it comes here, this pump stops working, and more sodium starts pumping in. Starts diffusing in. There are some voltage gated channels also. So more sodium influx takes place. Now because of more sodium influx, what happens? The membrane on the inner side starts becoming positive because this is a cation. So the membrane starts becoming positive. Is that clear? Now this happens only in one part of the neuron which has received the stimulus. The rest of the membrane is still normal, that is negative on the inner side, and it is negative on the outer. So now what was negative on the inside has become positive. Now this is called depolarization. This is called, the poles have been reversed. So this is called depolarization. Now, how does depolarization occur? It is because majorly of the sodium influx. Please tell me these points are clear. It is majorly because of the sodium influx. And what is the potential here? It is plus 30 millivolt. Is that clear? Now, since lot of sodium is getting pumped in, so more sodium is coming in, this sodium moves into the next part of the axon as well, right? Which changes the situation here. Now after a while, what happens? This part, this depolarization is going to flow to the next part, and from here, so let me draw it here. So in the initial part which got depolarized, so what will happen? Now earlier sodium was coming in, now later on potassium will start going out. So in that region, in the region where depolarization occur, later on potassium will start going out. So because of the potassium efflux, guys, your repolarization is going to take place. Is it clear? And the sodium which has moved into the next part, it will cause the sodium influx to occur in the second region, and so depolarization will flow from this part to this part. So this part becomes depolarized and the initial part becomes repolarized. So it will become repolarized to minus 70 and even to minus 90 millivolt, and then your sodium potassium pump will become active again. Please tell me these points are clear. Please tell me these points are clear. Is that clear? So see, it says, what does it say? So immediately the axon site B has a positive charge, outer surface, and negative charge at the inner side. So this is how the polarity or at the site is reversed and action potential is generated. So impulse, what is impulse? It is the action potential generated at site A which arrives at site B, and this is how it moves on and on. So basically the site is a stimulus, the rise in the stimulus induced permeability of sodium is extremely short-lived, quickly followed by a rise in the permeability for potassium. Is that clear? So that is how the membrane goes on getting depolarized from one place to another. And now once it has reached the end, it has to be transmitted to the next neuron. So this is called transmission. So it is from one neuron to the another through junctions known as synapse. So one synapse, guys, is made up of three components: presynaptic neuron, postsynaptic neuron, right, and a gap known as the synaptic cleft. These are the three parts of neuron. Is that, oh sorry, of the synapse? 1, 2, 3. Now, the synapses are of two types. One is electrical synapse, second is chemical synapse. Now, chemical synapse will involve a chemical, that is a neurotransmitter, while electrical synapse is like two wires connected with a third wire. Is that clear? So see, it is saying electrical synapse, the two membranes are in very close proximity. So current can generally flow directly from one neuron to another across the synapse. Is that clear? So this synapse is very similar to the impulse conduction along a single neuron, and it is always faster than the chemical synapse. But these are also rare in our body. The major synapse that we have in our body is the chemical synapse. Why do we call it chemical synapse? Because it is going to use the chemicals called neurotransmitters. So basically there is a gap. So the pre and the postsynaptic membranes are separated by a fluid-filled space called synaptic cleft. Is that clear? So as soon as the message reaches, see, as soon as the message reaches this presynaptic membrane, it is going to open the calcium channels. So calcium starts infusing into it. Now there are vesicles present, synaptic vesicles, which contain the neurotransmitters. This calcium moves in and binds to these synaptic vesicles, and these synaptic vesicles start fusing with the presynaptic membrane. Are you getting that? And all these neurotransmitters will be released in this space, which is called the synaptic cleft. This is the synaptic cleft. Is this point clear? Now from here guys, there will be another neuron, which is the membrane of next neuron. We call it postsynaptic membrane. Is this clear? Now these will have receptors for the neurotransmitters, and this is where the neurotransmitters are going to bind. And this means the transmission of impulse from one neuron to another. Please tell me that these points are clear. Yes, guys, please tell me these points are clear. Quick. Is this topic important? Yes, why not? They can easily give you this question in the sequence format. Yes, easily give you this question in the sequence format. All right then. So this is the diagram which NCERT is given you. So this is the axon terminal. These are the synaptic vesicles. This membrane is the presynaptic membrane. Then the space is synaptic cleft, and the next neuron membrane is called the postsynaptic membrane. Is that clear? And these are the receptors. So where are receptors present? This is also a PYQ. So receptors are present on the postsynaptic membrane, and these three components constitute a synapse. Is that clear? Now, since there are so many activities involved, the influx of calcium binding to the synaptic vesicles, then release, and then binding to the membrane. So, of course, this synapse is slower. This is slower. Now, see, it says the neurotransmitters bind to the specific receptor. So, for every different neurotransmitter, we have a different receptor. The binding opens the ion channels, allowing the entry of ions, which can generate a new potential in the postsynaptic membrane, and the potential can be either excitatory or inhibitory. Now, that depends upon the type of the neurotransmitter. That depends on the neurotransmitter. Now, very important, coming on to the central nervous system, we have brain, which is the main part of the CNS. Other than brain, it is a spinal cord. So starting with the brain, which acts as the main command and the control system, controls both voluntary movements, balance of the body, functioning of vital involuntary organs as well, thermoregulation, hunger and thirst, the circadian rhythm of the body, several endocrine glands, human behavior. So it is a site for processing of vision, emotions, thoughts, intelligence, hearing, speech, memory, etc. So there are so many diverse functions played by brain. Is that clear? Now, like many other vital organs, brain also has well protected coverings. It is protected by, number one, the skull, and particularly the brain box or the cranium. Right? Brain box or the cranium made up of how many bones? 8 bones. Clear? Then it has protective coverings called cranial meninges. Now, these cranial meninges are 3 in number. We have 3 meninges. Frog has 2 meninges. Can you let me know the answer in the chat, guys? We have already discussed frog. Which meninges out of these 3, which meninx is not present in frog? So we have duramater, we have pyometer, we have arachnoid, right? But which meninx is not present in the frog? Yes, this question has to be answered by you. So the meninges, they are in the format DAP. We remember it? DAP. This is outer, this is middle, this is inner, and then we have the surface of brain. So pyometer is in contact with the brain, and there we have skull. So duramater is in contact with the skull. Is that point clear? Please tell me, is this point clear? So see, outer layer is duramater, and a very thin middle layer which is called arachnoid, and then inner layer that is in contact with the brain is called the pyometer. Is that clear? So let me know which one is not present. Very good, Sankal, very good. It is arachnoid. It is arachnoid. So by the way, guys, in between these there are some spaces present. So below the duramater we have subdural space, and then below the arachnoid we have subarachnoid space. Now both these spaces are filled with different fluids. Is that clear? There are different fluids. This is cerebrospinal fluid, this is another fluid, not the cerebrospinal fluid. Is that clear? So this is the brain surface. Now the brain is further divided into 3 parts, that is forebrain, midbrain and hindbrain. Let's see the diagram. So the forebrain, first of all, consists of cerebrum, thalamus and hypothalamus, which constitutes your diencephalon, and that's it. Okay, these are the parts. So cerebrum is further divided into 2 cerebral hemispheres, which are connected through corpus callosum. Cerebrum is the largest part of your brain, yes, and do we see it is highly convoluted, highly folded? Why folded? To increase the surface area so that there are more neurons. Is that clear? Now, this is cerebrum. This is the largest part of the brain, and it is further made up of 2 cerebral hemispheres connected through corpus callosum. Now, these 2 parts constitute your diencephalon. Second part is the diencephalon. This is 1, this is 2. Is this clear? So hypothalamus and thalamus. So this is the thalamus, this is the hypothalamus, right? Now this thalamus, there are two thalami. There are two thalami, basically. One thalamus, right? Diencephalon. And here we have the third part also, which is the olfactory lobes, which is not mentioned here. So this is the third part of the brain called olfactory lobes. Clear to all? And then we have midbrain. Now midbrain is going to connect the different parts of the brain. And then we have the hindbrain. We have the hindbrain. Hindbrain is further made up of pons spheruli, cerebellum, and medulla oblongata. So first of all, you should know the different structures present in each part of the brain. Are these points clear? You should know the labelings. Now let's read the structure. So as told you, forebrain has cerebrum, it has thalamus, it has hypothalamus. So these two will constitute the diencephalon. Cerebrum forms a major part of the human brain. Longitudinally, it is divided into two halves, which are called the right and the left cerebral hemispheres, which are connected by a tract of nerve fibers called corpus callosum. Okay, they are connected by a corpus callosum. Is it clear? Now, the outer part of the brain will be known as cerebral cortex with prominent folds, I told you, to increase the surface area, which is known as the gray matter. Cortex is known as the gray matter. Clear? Due to the grayish appearance. Now, why is it gray? It is gray because of the presence of cell bodies, which are so concentrated here. So, there are three parts: motor areas here, sensory areas, and the large regions which are called the association areas. So basically, see, this cortex has, number one, or gray area has motor area, it has a sensory area, and it has the association areas. Now all these areas are going to play different roles. See, sensory, the most important is the association area, not important for brain, but important from examination point of view. In association areas for complex functions like intersensory associations. Intersensory associations, memory, communication, all these are going to be due to your association areas. Are you clear? Now there are fibers of the tracts covered with myelin sheath, which constitute the inner part of the hemisphere, which is known as the white matter. So myelin sheath is white in color, so it will give it white color and it constitutes the white matter. It is opaque. Are these points clear? So there are two parts, the gray matter and the white matter. Gray matter on the outer side and white matter on the inner side. Now cerebrum wraps around a structure called thalamus. Is it clear? Which is a major coordinating center for sensory and motor signaling. This is the function of thalamus. Another important part is the hypothalamus, which lies at the base of thalamus. That is why we call it hypo. Hypo means below. So guys, there are number of centers here for body temperature, urge for eating, drinking, and also several neurosecretory cells, which becomes endocrine in nature. These are endocrine in nature, secrete hormones. See, it's written. Is it clear? The inner part of the cerebral hemisphere, deep-seated structures like amygdala, hippocampus, now they constitute the limbic system of the brain. Now limbic system is associated to your emotions, so we also call it emotional brain, and the sexual behavior. Along with hypothalamus, so hypothalamus, what are the functions of hypothalamus here? Functions of hypothalamus: eating, drinking, then endocrine, then temperature, then also sexual behavior. So sexual behavior is controlled by hypothalamus as well as your limbic system. So please see if these points are clear to you. Please see all these points are clear to everyone. Yes, please. Is that clear? Okay. Now, moving on to the midbrain. See, The midbrain, as said, midbrain is between the thalamus or hypothalamus and of the forebrain and the pons of the hindbrain. So this is the location, and and through the midbrain or behind the midbrain actually is present a cerebral aqueduct that passes through the midbrain. Okay. Now dorsal part of the midbrain. So basically, if this is the midbrain, the dorsal part has 4 optic lobes. Frog has 2 optic lobes, and these 4 optic lobes are also known as corpora quadrigemina. Quadra means 4. Is that clear? So there are 4 round swellings known as corpora quadrigemina or 4 optic lobes. So the basic function of midbrain is to connect various parts of the brain. Then we have hindbrain. So one, pons feruli, then we have cerebellum, and then we have medulla. Now pons, as we've read in the respiratory system also, they are fiber tracts that are interconnected, and they are going to pons, they're going to control your respiration. So respiratory centers. Do you remember pneumotaxic center? Pneumotaxic center, which is known as the switch off center. Pneumotaxic center, that is present here. Clear? Now cerebellum, it says cerebellum has a very convoluted surface just like your cerebrum, and for accommodating the most neurons, of course. And then medulla. Medulla is the part of brain which is connected to spinal cord, and it controls the involuntary processes. So medulla oblongata, it controls mostly the involuntary processes. Is that clear? It controls the involuntary processes. Please tell me all these points are clear. Involuntary processes. Now this is an important line. So brain stem. What is a brain stem? So brain stem, sorry. So see, brain stem is formed of midbrain, pons feruli, and medulla oblongata. So in case they have written brain stem is formed of midbrain and hindbrain, it is not exactly right. Right? Because in hindbrain, the cerebellum part is not a part of brainstem. Is that point clear? So brainstem forms a connection. So basically it forms a connection between the brain and the spinal cord. Please tell me that these points are clear to you all. Please tell me all these points are clear to you all. So see, these are the differences. Now I'm going to move on to the last chapter of human physiology. So these differences between neural system and endocrine system I've just discussed, that endocrine is long-lived, the effect of neural system is short-lived. Do you see? All these points have been discussed already. Now talking about the endocrine glands. First of all, what are endocrine glands? These are ductless glands. These are ductless glands, and their secretion is known as hormone. What is hormone? The chemical produced are released into the blood. It is through blood that hormone is going to move, transported to the distantly located target organ. Now, what is the proper definition? It is a non-nutrient chemical, acts as an intercellular messenger produced in trace amounts. Very little amount of hormone is needed. Please tell me these points are clear. So these endocrine glands have been present in invertebrates also, but they have been very fewer and very simpler as compared to those present in higher organisms. Let's talk about human endocrine system now, finally. Let's buck up, guys. Let's have a quick break. Thumbs up. Yes, please. All here with me? All here with me? Quickly let me know. Ma'am, pons also records the circadian rhythm? No. No, no, no. Not the circadian rhythm. It is a pineal gland. We'll talk about it now. All right, guys. So human endocrine system. So there are different glands that we're going to talk about. And in addition to these, there are some other glands also like GIT, liver, kidney, heart, which also produce hormones. We'll be discussing them one by one. Let's talk about the hypothalamus. Now see, hypothalamus we have talked about in nervous system also. Hashtag, it is a connecting between, it is a connection or connecting link between neural and endocrine system. And endocrine system. So it is a connecting link. So we have seen that it is a thermoregulator, it has a drinking and eating center, right? Satiety center, hunger center. But now here we'll see that hypothalamus can also produce hormones. So where is it placed? I already told you it is placed below the thalamus. Now they've not used the word diencephalon there, they've used it here. So thalamus, hypothalamus, they together constitute diencephalon, and this is where it is present at the basal part of diencephalon, that is below the thalamus or forebrain, right? What is the functions? Number of functions. There are several groups of neurosecretory cells, we call them nuclei, which produce hormones, and these hormones regulate the synthesis of and secretion of pituitary hormones. The hormones produced by hypothalamus are of two types. Let's see what are the two types they are talking about. One is the releasing hormone, right? Another is the inhibitory hormone. The name itself says releasing hormones are going to stimulate the interior pituitary. See, and which pituitary? Not any, but interior pituitary basically. And inhibiting are going to inhibit the secretion of interior pituitary. For example, they are saying gonadotropic releasing hormone, GnRH, stimulates the pituitary synthesis and release of these. Now we know that. And remember, it is not any pituitary, it is not the posterior, it is the interior pituitary only. Are we clear here? And similarly, another hormone, this is somatostatin. Somatostatin, guys, is another term for growth hormone inhibitory hormone. This is somatostatin. So what is it going to do? It is going to inhibit the release of growth hormone from anterior pituitary. Are we clear till here? So the point is, how are these hormones going to reach to the anterior pituitary? So basically they pass through axons and released from their nerve endings. These reach the pituitary through a portal circulatory system, which we have already discussed in the previous class. This one is known as hypophyseal portal circulation. Is that clear? So which is between, this particular circulation is between hypothalamus and anterior pituitary. And anterior pituitary. Please tell me these points are clear. Under the direct, while the posterior, it says the posterior pituitary is under the direct neural regulation of hypothalamus. And we know posterior pituitary is not going to produce any hormone on its own. No hormone of its own. It only stores and releases the hormone which is produced by hypothalamus. Please tell me all these points are clear. Yes or no? All these points are clear, right? Okay, now moving on to the first, that is the pituitary gland, which is also known as the master gland, right? If this is the master gland, then your hypothalamus, guys, was the supermaster. This was the supermaster, because we have seen this supermaster will either inhibit or make the anterior pituitary release hormones. So we have talked about the supermaster. Now we are going to talk about the master gland. Now see, it is located in a bony cavity called sella turcica and is attached. Now this sella turcica, this bony cavity, is of sphenoid bone. Remember, the base of the brain is made up of sphenoid bone, and there is present a depression, sella turcica, where this pituitary is located. Is that clear? It is attached to the hypothalamus through a stalk. So like this. This is posterior. So this is hypothalamus, this is anterior pituitary, this is posterior pituitary, this is the intermediate lobe, and this is the stalk called infundibulum. Is that clear? This is the stalk that they are talking about. Is that clear? Yes or no? I hope there's no doubt about it. Now it says it is divided anatomically into adenohypophysis and neurohypophysis, and adenohypophysis further has two parts, pars distalis and pars intermedia. Now pars distalis is known as anterior pituitary, right, and produces these hormones, growth hormone, prolactin, thyroid stimulating hormone, adrenocorticohormone, LH, FSH, and yes, these are the hormones. So adenohypophysis is the term given for anterior and intermediate. This is adenohypophysis, right? While this posterior pituitary is known as neurohypophysis. Please tell me if these points are clear to you all. Right? This is adeno. And then, by the way, anterior pituitary is also known by the name of pars. Sorry. It is also by the name of pars distalis. We call it pars distalis. And this one is known as pars intermedia. While posterior pituitary will be known as pars nervosa. Please tell me if these points are clear. Yes, these points are clear to all. So these are the hormones. And then pars intermedia also produces a melanocyte stimulating hormone, almost merged. This pars intermedia is almost merged with the pars distalis. Now talking about neurohypophysis or the pars nervosa. So wait a second, where is it? Yeah, pars nervosa. Now, over-secretion of growth hormone. Let's talk about this. Growth hormone stimulates the abnormal growth of the body. The name itself says we are talking about anterior pituitary, guys. Hormones of anterior pituitary. Growth hormone will cause abnormal growth of the body, leading to gigantism. And lower secretion will cause stunted growth, and this is known as gigantism. So, low and over secretion. These are the two disorders being discussed. Now, if this is produced, excess secretion in the middle age will lead to acromegaly. Severe disfigurement of the body can lead to serious complications, even premature death. This is also known as the gorilla-like appearance. There is gorilla-like appearance here. Now, disease is hard to diagnose in early stages and goes undetected. So, yeah, that is growth hormone. Low, dwarfism. High, gigantism, but if high in the middle age, then acromegaly. Then talking about next hormone, which is prolactin. Prolactin is for mammary glands for the formation of milk. Then we have TSH, it will stimulate the thyroid gland for releasing the thyroid hormones. Are these points clear? Then we have glucocorticoids, guys. Glucocorticoids, secretion of state. We have corticoids, adrenocorticoids we call them, adrenocorticoid ticotropic hormone, which is ACTH. ACTH stimulates the synthesis of steroid hormones from adrenal cortex. Okay, so ACTH from anterior pituitary is going to stimulate the adrenal cortex to produce different types of glucocorticoid, mineralocorticoid, 6 corticoids, all the corticoids. Then we have gonadotropins. These are of two types, FSH and LH. Are these points clear? Now, what will happen? In males, LH stimulates the synthesis and secretion of androgens from testes, and in males, FSH and androgen regulate the sperm production. While in females, guys, LH induces the ovulation. We already know LH surge and maintains the corpus luteum formed from the remnant of graphene follicle. FSH is important to stimulate the growth and development of ovarian follicles. We'll read more about these gonadotropins in detail in human reproduction. While MSH coming from pars intermedia, it acts on the melanocyte cells, right? Melanin producing cells, and therefore it regulates pigmentation. Oxytocin, guys, oxytocin and ADH, these are produced by, see here, ADH and oxytocin or vasopressin and oxytocin, they are produced by, they are produced by hypothalamus, right? Produced by hypothalamus, and what? Produced by hypothalamus and secreted by, and released by posterior pituitary. posterior pituitary. Are these points clear? Right? So what are the functions? We have already read about the function of ADH, right? What does it do? And let's talk about oxytocin. It acts on the smooth muscles and stimulates the contractions, vigorous contraction. So it is known as the birth hormone, right? And it also causes milk ejection. So it is known as the milk ejecting hormone. While if we talk about ADH, so ADH is for reabsorption of water and electrolytes from DCT. Remember? The conditional reabsorption that occurs by ADH. So it causes, it basically reduces the loss of water. So it reduces diuresis. It is against diuresis. Now see, in case there is impairment and excessive release of ADH, less release of ADH, then what will happen? There will be excessive loss of water resulting in dehydration, and this will be known as diabetes insipidus. So this happens when ADH is low. Low ADH will cause diuresis. Low ADH will cause excessive urination, that is diuresis. It will cause dehydration, water loss. This is diabetes insipidus. Are these points clear? Now moving on to the next gland, which is the pineal gland. This is present on the dorsal side of the forebrain, okay, and releases melatonin, very important role in maintaining the 24-hour or the diurnal rhythm of the body, which is also known as circadian rhythm. This is also known as circadian rhythm, biological clock or biological clock. Clear? You can call it sleep-wake cycle, body temperature, normal rhythms, all are controlled by melatonin. They also influence the metabolism, pigmentation, defense capability, and even the menstrual cycle. So that is the function of pineal gland. Now moving on to the thyroid gland. So thyroid gland has two lobes basically, and present on each side of trachea. They are interconnected by a thin flap of connective tissue, which is known as isthmus. So say if this is thyroid gland, right, and this is the isthmus. This is the isthmus. So there are two lobes, and now each lobe is follicular. Each lobe is follicular. So there are two types of cells basically. So there are two types of cells. One are follicular cells, which are going to produce T3 and T4, and there are other cells called parafollicular cells, which are going to produce another hormone called TCT. So these are the three hormones produced by your thyroid gland. Three hormones produced. Now see, thyroid gland is consisting of follicles and the stromal tissue. Each follicle has follicular cells, and these follicular cells will form tetraiodothyronin, that is T4, which is actually called thyroxine, and then triiodothyronin, that is T3. So T3 is converted into T4 and the vice versa. Is that clear? And iodine is essential for the formation of these two hormones. So these hormones require iodine, not TCT. Is that clear? Now deficiency of iodine leads to hypothyroidism. So if there will be deficiency, low iodine, low T3 and T4. Now this results in enlargement of the thyroid gland, and this enlargement is known as goiter. Is that clear? Now hypothyroidism during pregnancy can lead to defective development and maturation of the growing baby, stunted growth, and this during pregnancy it can lead to cretinism in the child. So mentally retarded, low growth, very weak, right? And also the baby born will be infantile sterility it leads to. So see, it is leading to mental retardation, low IQ, abnormal skin, dead mutism, and then in adult women, hypothyroidism can cause menstrual cycle to become irregular. And we have seen this. So many girls suffering from hypothyroidism, they have disturbed menstrual cycle. Now, another situation when there is cancer of thyroid gland. Now, this will lead to increased thyroid hormones will increase. This is known as hyperthyroidism. So, it is just the reverse of hypo. Now, it also occurs due to exophthalmic goiter. This is also another type of hyperthyroidism in which there is enlargement of thyroid gland, production of eyeballs, increased BMR, weight loss, and also known as Graves' disease. By the way, this is autoimmune disorder. This is autoimmune in nature. Please tell me these points are clear. Now, what are the basic functions? It is thyroid hormones. They are involved in regulating the metabolic rate, and then RBC production, metabolism of all fats, carbohydrates, and proteins, maintenance of water and electrolyte balance. Also, yeah. So when it comes to BMR, they also regulate your heart rate, your breathing rate, your blood pressure. So all these things. And now coming on to another hormone, thyrocalcitonin, TCT. So the name itself says it is going to regulate the calcium level. So basically, how does it do that? So TCT is going to decrease the blood calcium level. It is going to decrease the blood calcium level. So we call it hypocalcemic hormone. It is hypocalcemic. How does it decrease the blood calcium? So basically, what is it going to do? It is going to send, so this is the bone, let's say this is the bone, and this is your blood, right? So it's going to transport calcium into the bones. So this is how blood calcium decreases. Is that clear? Also, it is going to release calcium in urine. So bone, calcium in bones. Calcium loss in urine, and feces. So this is how, and absorption of calcium also decreases, lowers the absorption of calcium through food. So this is how it decreases the blood calcium level. Are these points clear? Then moving on to the another gland, which is parathyroid gland. Now parathyroid gland is present on the dorsal side of thyroid gland. Say this is thyroid gland, and on the dorsal side, on the back side, on the four corners, guys, we have these four parathyroid glands. So 4 in number. On the back side of thyroid gland, one pair in each of the two lobes. Is that clear? And it is going to produce this hormone, parathyroid hormone. This is just the opposite of TCT. Yes, it is antagonistic of TCT. So like we have seen that TCT is, see here, TCT is sending TCT is sending calcium into the bone. This is going to send calcium back. This is PTH. So basically, PTH is going to cause demineralization of the bones. See here, it increases calcium level in blood by process of bone reabsorption, stimulates reabsorption calcium from renal tubules, and increase calcium absorption from the digested food. So it is a hypercalcemic hormone, and they together maintain the role of calcium balance in the body. Are these points clear? Then we have thymus, guys. Thymus is a lobular structure present between the lungs behind sternum on the ventral side. The main function is the development of immune system by secreting hormones called thymosins. Now these thymosins will cause differentiations of T lymphocytes. Remember, there are 4 types of T lymphocytes: TH, TS, TK, and TM. So they are because of thymosins. Also, they promote the production of antibodies. How? Because you know that it is the T helper cell which stimulates the B cells, right? So T helper cells will stimulate the production of antibodies and therefore humoral immunity. Is that clear? So it degenerates in old people and therefore results in decreased production of thymosins and their responses become weak. Are these points clear? Then moving on to the adrenal gland. Now very, very important. So adrenal gland, one pair of adrenal glands, one on the anterior side. Now this is a wrong statement. This has been changed. It is written one above each kidney, right? So we know it is present above kidney. So that is why we call it suprarenal. That is why we call it suprarenal. So this anterior word has always been wrong. In case you are using that book, please change. Now, the gland is composed of two tissues. Outer is known as cortex. This is the outer, this is the inner. So let's say if this is the medulla, this is present on the kidney. So this one is the adrenal cortex. This is the cortex, and this one is called the medulla. Is that clear? Now, different regions, they are going to produce different types of hormones. Now, this cortex is going to produce corticoids, while the medulla is going to produce catecholamines. Now, as the name itself says, these are amino acid derivatives. These are steroid in nature. Is that clear? Now, it says underproduction. So, it is underproduction leads by the adrenal cortex leads to Addison's disease. It is the underproduction which causes the Addison's disease. This is the diagram that I've just told you. And then talking about the hormones. Adrenal medulla, as I told you, they release these two hormones, adrenaline, also known as epinephrine, noradrenaline, also known as norepinephrine, and together they are known as catecholamines, or we call them emergency hormones or hormones of flight. So what do? They increase the alertness, pupillary dilation. You have just felt the effects on 3rd May, and every day NTA is giving us adrenaline rush, right? So alertness, pupillary dilation, piloerection, sweating, all these are because of catecholamines. So these both, they increase the heart rate, strength of heart rate, contraction and rate of respiration. They stimulate the breakdown of glycogen because they are preparing us for fight. So glycogen is a stored form of food. We need more energy. So glycogen will be broken down, resulting in increased concentration of glucose in blood. So it also stimulates the breakdown of lipids and proteins. Both of these will help in increased production of energy. Please tell me, increased production of energy. Are these points clear? Yes or no? Then we have cortex, guys. Cortex can be divided into 3 layers. Now, let me tell you how cortex is divided. So, if this is the gland, this is the medulla we have discussed. Now, I'm going to divide the cortex. Outer, middle and inner. This outer, middle and inner. Right? The outer one it says is known as the zona glomerulosa. Zona glomerulosa. Right? This is the zona fasciculata, middle one. And then the inner one is zona reticularis. Is that clear? Is that clear? This is the zona fasciculata, and then this is the zona reticularis. Now, all of them, they produce different hormones. Glomerulosa is known to produce mineralocorticoids. Is that clear? See here, cortex secretes many hormones. Together we call them corticoids. And then corticoids, this is glucocorticoids, which are involved in this corticoid balance spasm. Okay, now let me tell you which ones are which. Fasciculata produces glucocorticoids, and reticularis produces sex corticoids. Now see, mineralocorticoids, the most important of all is aldosterone. And we have seen the function of aldosterone in RAS mechanism. It helps in sodium reabsorption, right? So it is involved in sodium reabsorption from DCT and CT. Is that clear? It is the main mineralocorticoid, aldosterone. Clear to all? Right? Then we have this glucocorticoid. The main glucocorticoid is, guys, cortisol. This is also known as the stress hormone. So see, glucocorticoid, the major one being particularly cortisol. What does it do? It inhibits the cellular uptake and utilization of this. It stimulates the gluconeogenesis, lipolysis and proteolysis, maintaining the cardiovascular system as well as kidney functions. Is that clear? It produces the inflammatory reactions. So whenever there is stress, cortisol is going to cause inflammation and suppress your immune response. It also produces RBCs. Aldosterone, as we have read, it acts on and causes reabsorption of sodium and water. Please tell me all these points are clear. Now, aldosterone is just a repetition. maintenance of electrolyte, body fluid volume, osmotic pressure, and blood pressure. Androgenic steroids. Now, these are the sex corticoids. These are the sex corticoids that we are going to talk about. Secreted from zona reticularis. Is it clear? So, which produces main role in the growth of axial hair, pubic hair, and facial hair during the puberty. Please tell me all these points are clear to all. Yes, guys. Let's have a quick break. Thumbs up break. Yes, please. Let's have a quick thumbs up break. Everybody, are you here with me? You're almost going to complete this. So pancreas is a composite gland because it is both exocrine and endocrine. The endocrine part is known as aisles of Langerhans, right? So 1 to 2 millions of aisles are Langerhans, and they produce two main hormones. So there are two cells, alpha cells and beta cells. So beta cells are going to produce insulin, while alpha cells are going to produce glucagon. Is that clear? So what is the function? So basically alpha cells, they increase the blood sugar level, increase glucose in blood. So we call glucagon as hyperglycemic. And insulin, this is known to decrease the blood glucose level. So it is hypoglycemic. Please tell me these points are clear to all. Yes, please. Right? So both of them, they are peptide hormones. This is what I have already told you. Moving on to, so this is discussed. Now, testes. Testes, a pair of testes will, although the details I'll be discussing in human reproduction. So see, it also has dual function. So one is, it is a sex organ, and second, it is endocrine. So the testes are composed of seminiferous tubules and stromal and interstitial fluid. And there are Leydig cells basically. Leydig cells are the interstitial cells which are known to produce testosterone. Is that clear? And they are androgens. Clear? And then we have the functions of androgens, that is development, maturation, and functions of male accessory sex organs. All these structures, they will develop because of this. Also stimulate the muscular growth, facial axillary hair growth, aggressiveness, low pitch, all this. And stimulatory role in spermatogenesis, also affect your neural system and influence the sexual behavior, that is libido. They also have anabolic effects on proteins and carbohydrates. That is why people take these steroid drugs. Now, females, again pair. So there are different structures producing different hormones like estrogen and progesterone. So there are follicles which are known to produce these different hormones. So before ovulation, growing ovarian follicles, they are known to produce estrogen majorly, and after ovulation, it becomes corpus luteum, which is known to produce majorly progesterone. Right? What are the functions? How do they support pregnancy? We'll be discussing it in human reproduction. Now, there are other structures also like heart, kidney, and GIT. So we have seen that heart is going to produce ANF, and we have seen the effect also. We have seen kidney producing renin, and there is another hormone being produced, that is erythropoietin, which is going to increase the RBC production. See here, so kidney, the same juxtaglomerular cells, they will produce erythropoietin, which stimulates RBC production. Now coming on to the different hormones of GIT. This is gastrin, secretin, CCK, GIP. These are the different hormones produced by your alimentary canal and the surrounding regions. So secretin basically, it acts on the exocrine part of pancreas and stimulates the secretion of water and bicarbonate ions. CCK has effect on pancreas and gallbladder, so stimulates the secretion of pancreatic juice and bile juice. So GIP, it inhibits the secretion, gastric secretion. And then there are so many other structures which produce the growth factors. Is that clear? Now, last not the least, we guys have mechanism of hormone action. So for mechanism, let's directly come on to this part. Now see, first of all, we have 4 categories of hormones. The hormones which are protein derivatives, the protein hormones, proteinaceous hormones, and amino acid derivatives. These hormones will show this type of hormone action. What is happening? So they have extracellular receptors, or you can say membrane bound receptors. They have membrane bound receptors. Is that clear? So hormone will be binding to this receptor present on the membrane. So since the hormone is not able to move into, so there is production of secondary messengers like these, which will cause biochemical responses and eventually the physiological responses. So this is for all the protein hormones and amino acid derivatives. While there are those hormones which are steroid derivatives. Steroid hormones and iodinated hormones, that is T3 and T4. For T3 and T4, guys, the hormones have the receptors inside the nucleus. So since the receptor, see, this is the receptor is intracellular. Receptor intranuclear or intracellular. Intranuclear, that means within the nucleus. Therefore, the hormone itself enters into this. Okay, so there is no second messenger, no secondary messengers required. Is that clear? So hormone receptor complex is formed within the nucleus, and this causes some genetic changes and resulting in the physiological responses. So guys, with this we complete human physiology. I'll be sharing these notes with you on my Telegram channel. And before I leave, I'm again telling you the Telegram channel is Tina Maam. There's no space, of course, biology. I'll be sharing my notes here after the class, and don't forget to join me tomorrow for plant physiology. Again, same channel, Unacademy NEET English channel, and we will be completing it from 7. We'll be we'll have class from 7 p.m. So tomorrow again we have a marathon where we are discussing now plant physiology. Very important because of the numericals, and then there's C3, C4 cycle, and then PS1, PS2. We'll be discussing those intricate questions also in the class. So don't forget to join me, and all those who are appearing in the Re-NEET exam, guys, make sure that you use this opportunity of taking a Prime membership for just ₹1, where you can watch all the recorded lectures, take this AIMT, and you can practice so many questions. Let's leave no stone unturned so that we are getting better marks. So it should like be a slap for the the failed system. So work hard, and I'm going to see you tomorrow at same channel, same time, that is 7 p.m. to discuss plant physiology. Bye bye, guys.